Well, good morning, everybody. Or as we say in Trinidad, morning. This is the first dive of the Deep Wonders of Trinidad and Tobago expedition, which is being operated by Schmidt Ocean Institute. We are on their incredible research vessel, Falkor 2, and we are just west of Tobago. And right now, we are at a depth of about 1,040-ish meters. And this expedition, as this is our first dive, I'll tell you a little bit about what we're going to be doing. My name, first of all, is Dr. Diva Aman. I am the chief scientist of this expedition. I am one of the directors of Species, a local NGO in Trinidad and Tobago that does ocean research, education, and advocacy. And I am just one of 25 scientists on board this incredible vessel from a range of different institutions. You will hear from them over the month that we are out here. We plan to be hopefully diving nearly every day from today to around the 26th of July. So we hope you'll join us. And this expedition is really about trying to understand what is in our waters. Trinidad and Tobago is more than 93%. The ocean is more than 93% deep waters. So that means below scuba depths. And because of how expensive the technology is to explore the deep sea here, we just don't have access to that. So this is a really incredible opportunity, and it is going to allow us to really understand what lives in our ocean, and therefore try to be able to steward it a bit better. So pilots, if you are willing, I think let's take a little mooch around for about 10 minutes if we head towards waypoint 2. And then the first thing we're going to do is test the heat flow when Rachel joins us. And so if we see anything interesting, we will stop to zoom in. But yes, so this is really about trying to understand our country a bit better and, of course, share it with all of you as well. This will be the first time that most people viewing the feed who are joining from Trinidad and Tobago will have ever seen the deep sea. We can see a nematocrustinid shrimp in front of us. If we do a quick zoom, they have some really long legs. And as you can see, we're in a really soft sediment environment. What we're going to be doing today is moving slowly uphill. Hill is a little bit of an exaggeration. It's more of a flank along the side of a canyon, if you will. Again, canyon is a bit of an exaggeration, but we expect there to be a lot of soft sediment and a lot of incredible animals like this shrimp. Okay, once you guys are satisfied with outreach images, we are happy to go on. And if there's anything that anyone in the back would like to see, don't feel bad to say. We're so happy for so many of you following along on YouTube and Twitch. Please do share, friends, family. We would like as many people as possible to be joining us. And we're seeing that we have people joining us from not just Trinidad and Tobago, but all over the world. So thank you for being here. This is a really landmark moment for Trinidad and Tobago. Yeah, so that looks like a sponge. Can we do a quicker zoom? No, let's keep going. Yeah. And so as I was saying, because deep sea exploration is so expensive, you need these incredibly expensive remotely operated vehicles or very large ships like we're on. It means that most countries in the world, for instance, small island developing states like Trinidad and Tobago, we just don't have access to that technology. And so having this opportunity of the research vessel Falkor 2 come to Trinidad and Tobago is really incredible and important. And really, we hope this is the beginning of what is locally led deep sea research in Trinidad and Tobago and the Caribbean at large as a result. Oh, a sea pen. Oh, so maybe it wasn't, the other thing wasn't a sponge, it was a sea pen. Oh, beautiful.
From the bottom. So at the base, there's
Can we have a quick lasers on just to get a gauge of size?
So it is fine. With this cable. So when it's zero, it's really zero.
So those lasers that have just come on, those two green dots you can see at the bottom of the screen, those are to help us to measure. They are 10 centimeters apart, so you can use those to estimate the size of the animals we're seeing. Thank you guys, that's great. We can move on, yeah.
You want to just leave the lasers on?
Oh, well, I was to test the heat flow. Are you guys ready to? Do we need Rach, or you're good to go?
It'd be better if she was here.
Yeah. Would you mind going to get her, and then once you guys are here, we can go.
Yeah.
Zach, is this slope okay for you guys to test the heat flow?
Yeah.
Great. Okay. So let's go ahead, and once we're joined by Rach and Rob, we'll sit down and do that. So on this dive, we're trying to, as I said, get an understanding of what is down here, get eyes on what is down here, and then take samples so that we can be able to try to analyze that after we get off the ship to, again, try to get a really good picture of what's down here. We're going to be doing a series of tasks on this dive. This site is 1A. We've gone for some really exciting naming conventions there. But so our goals are to really observe what's down here. We're going to be doing a series of video transects. So that means you can watch and we can watch and we can see all the things. Well, on those transects, we are going to be looking for evidence of seepage, so fluids or gas seeping from the floor, because that's often the indication of high density animal ecosystems. We're also going to be looking for high coral and sponge density, which, looking at the seafloor now, that's probably unlikely given this is a soft sediment environment. In addition to those video transects, we're just going to be observing animals, looking at their behavior, capturing imagery of it, and also sampling them. Because we don't have the technology, it means that we really don't have a great understanding of what is in our waters. There was a study published by one of the scientists on board. She's a student at the University of the West Indies just a few weeks ago, Jamie Lee Luchin. Please do look that up. But essentially, she went through and, can we get a zoom on this halosaur quickly? So here we have a halosaur fish. They're really easy to identify to the family because of that characteristic straight body and then the tail at the end that is their propulsion. And these are very common a very common family of deep sea fish. Great, thank you guys.
Very cool name.
Very cool. So as I was saying, this study found they did an assessment of all of the records of any species that had been observed or sampled from Trinidad and Tobago's deep sea since sampling had begun, and that was in the 1950s, and there's been a series, just a handful of expeditions, very, very limited, and they found that about 451 species had been recorded so far. But what was incredible was that of these 451, she was able to do modeling that showed that if we were to keep sampling, which we are doing on this expedition, that number is likely to get up to as high as 1,600 species. And so that means about 70% of the deep sea species in Trinidad and Tobago's waters have not yet been recorded. And it's highly likely that some of those will be species new to science. Where we are diving today, no one has ever seen this part of the seafloor before, which is incredible. And that means, again, that we expect to see perhaps species that may be new to science, or potentially new to science, I should say, ecosystems that are potentially new to science. When you're working in the deep sea, that's a paleodicton down there, isn't it? the weird holes on the seafloor. Never mind. It's okay. There's already a lot to see. I can't remember what I was saying now. But in front of us we have another halosaur, and then what looks like, can we have a quick zoom on the anemone? This is a Venus flytrap anemone, right? Ooh. Looks like anemones on anemones. You guys ready?
We just need power, and then we can get ready.
Do we do power before we set down?
Yeah.
Yeah, just for us to
Right.
get a bottom water temperature.
So in addition to collecting samples so that we can try to understand the animals that live here, work out what species they are and whether they are new, we're hopefully going to be collecting some sponges so that we can do some eDNA work them. Ooh, a squid on the seafloor. And a ray. And a ray?
Is it a ray? What?
Maybe not. No. But the squid is cool enough. Let's go see the squid.
Or squid.
I'm a little tired.
There it goes.
Ooh. Oh, it looks like his tentacles, arms are in the
Changing colors.
sediment. Do we think it's that he's got some prey in there, or do we think he's just holding on?
It's holding on.
Or she? He or she? Or they? Oh, he's definitely got.
I thought maybe it was
Oh, he's trying to cover his eyes.
Too bright.
Do you want me to sit down here if we're going to sit down anyway?
Yes, that would be great. We'll sit down. When you sit down, you have a chance. Rach and Rob would like power to the heat flow. Oh, and is that a plastic bag? Someone's logging.
We were just asked for this.
Who's our logger?
No, we're longer for the first time.
No, that logger is you.
The geophysicist is logging.
Okay. Can we zoom in any more?
Oh, I'm going to sit there.
Oh, sorry. Sorry.
It's a
On
dude.
bottom.
Oh. Oh yeah, he's beautiful. Look at those eyes. He or she is beautiful.
Oh my god.
Can we follow him?
No. While we're sitting down, let's do the heat flow test.
Okay.
So we're also going to be collecting some samples for eDNA. That includes both water and animals. And then we're also, while on this expedition, we're going to be looking at the heat flow coming from the subsurface of the seafloor. And so right now we're going to, even though we're not doing heat flow measurements, we are going to be doing a test of the heat flow. So you're going to see, if you were to look in the back camera, you'll be able to see it. Rach, do you want to come and sit here and tell a little bit about heat flow?
Oh. Sure.
Great.
Yep.
So we're going to hear from Rachel Lauer, who is the scientist in charge of the heat flow. I'll be back. Oh.
Nope.
I know. Okay, the squid is wants to be with us, wants to be on camera.
Oh.
Look at those chromatophores, beautiful. So fascinating that he or she is just
Showing off.
very
Stressed.
present.
They're going to attack the sun. My turf.
Yeah, maybe that's some kind of defensive maneuver. Okay, right?
Okay.
All right, so we are ready. Just be aware, it looks like we're going to have to adjust our camera a little bit.
Yep.
Because it doesn't quite show the bottom.
Yep.
So are you comfortable with still doing that?
Yeah.
You see it in the bottom left there?
Yeah. I think you mean right there in front of, on the right hand side?
The bottom left on the quad there?
Okay.
What's the pointer? So the tip of your spear would be probably
Okay.
here.
I see. Yeah, I mean, we can adjust that.
I can do it very gingerly.
Yeah.
So 37%, so we gotta go here, Cal 6.
All right, so yeah, as was mentioned by Diva, my name is Rachel Lauer from the University of Calgary, and I'm here with Robert Perrin, my postdoc, who's getting set up for our first test of our heat flow probe. And what this probe is measuring is how much heat is coming out of the ocean crust and through the sediments. And that information is useful for us because we are studying primarily fluids and how fluids move between the ocean and the ocean crust. And that is interesting to this crew of mostly biologists because how fluid moves in the crust can help inform where communities thrive and where we may locate hotspots of biodiversity. So in this particular region, we expect most of our fluid flow will be coming from deep within this system, potentially flowing back into the ocean through structures like mud volcanoes or faults that provide these permeable pathways for fluids to make their way from deep below the surface, where they get warmed up and then make their way back to the seafloor.
Power is on.
All right.
We're ready to lower when you guys
Okay, so are you okay using Excalibur prior to this to make sure we have enough sediment?
are. Sure.
Yeah. Because
Alright,
although we
so-
see soft sediment, we just want to make sure it's not like a thin layer and then there's a hard crust.
Okay. So I'm gonna rack in.
Sorry, I should have mentioned that prior. Yeah, so one of the things we do prior to making a heat flow measurement, we have the heat flow probe mounted onto the side of the sub, and it's on a hydraulic ram that moves vertically and allows us to penetrate the sediment. But before we do that, we want to make sure that the sediment is soft and there's not a hard crust right below the surface, because we can break the probe. Despite it being very strong, the sub is stronger.
So you see them to sit there. That monkey's fist right there.
Yeah, so we have a pokey stick that has been named Excalibur for obvious reasons, that we're going to test the soft sediment and make sure we can safely conduct a measurement. And don't worry, we'll get back to biology as soon as possible.
More starboard, I think. Starboard and down. More starboard. There, touch me.
So this is really the only tool I think that requires power from the sub. Most of the people here are interested in the biology, which is obviously a big part of this cruise. We are going to be doing push cores as well, as someone had asked in the chat. Not on this dive, but on the next dive we will definitely be doing push cores. Also measuring temperature, ambient temperature, and temperature if we see any seeps or other evidence of flow. can also sample water near any sponges for things like eDNA. All right, that looks very soft.
So those are 5 centimeters in those tapes.
Cool.
Yep. Just want to make sure it's soft.
That looks good. Cool. Yep. So they're using this to make sure that we can safely use the heat flow probe, and they've taped off every 5 centimeters to be able to see to what depth we can actually make our measurements. And
What's your basket? Let's do basket. Big basket.
you probably won't need to begin this for this dive.
Good job.
We are good. Yeah. It's just setting up.
I'm recording right now.
I'll go, Anna. I'll go till 10:30 and then you'll take over.
Okay, you all ready?
Great. Thank
Okay,
you, pilots.
so
Yep. So we can.
We just got our bottom water temperature, and now we're gonna get a measurement.
So we just stay here for a bit more.
Yeah, sorry.
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 What was
Should we
you
go?
left?
Okay. I was just trying to give you back the hot seat.
All right, so you happy to lower?
Yes.
Sorry.
So this is really important. Thank you, Rach. This is Diva Amon again. So this is really important, this testing at the beginning, just so that we can have all subsequent dives be as smooth as possible. This is a piece of equipment that we're going to be using fairly frequently. And just a few moments, we should be all wrapped up, and then we can head off
Awesome.
to explore.
So we're gonna set it for 480 seconds or 600.
600? Yeah. Is that how it was before?
With the dynamic ranges in there. There's not one. You see when the heat pulse goes.
Yep.
So that's going to count down 600 seconds from the heat pulse.
And so while we're waiting for this to happen, I'll tell you a little bit about the geology of Trinidad and Tobago. I will caveat this by saying I am absolutely not a geologist. I'm a deep sea biologist, so not a great start. But Trinidad and Tobago sits in a really unique position. We sit on this massive seafloor feature called the Barbados accretionary prism. Most of our deep waters does. It may be called Barbados accretionary prism, and yes, Barbados is on it, but also a lot of the Caribbean is on it. And what that is when
No.
one of the plates is subducting below the other. There is a lot of off-scraping during that subduction, and that off-scraping combines with organic matter and lots of other things, and that is actually what creates the oil and gas that Trinidad and Tobago's economy and also Venezuela's economy actually survives on, or thrives on, I should say. And associated with that oil and gas as well, you can get really unique deep sea ecosystems. They're called methane seeps. And these are areas where methane and other chemicals seep out of the seafloor, and they power ecosystems when they do that. Down in the deep sea, there's not a lot. There is, I should say, not a lot. There is no sunlight. And so animals usually depend on food from the sea surface that have drifted down, like these pieces of sargassum you can see in front of the ROV, these brown pieces of sargassum. This is what animals eat. Bits of plankton that have drifted down. Marine snow is what we call it. But then, of course, you can get bigger parcels like dead whales, dead turtles, dead large fish, etc. And that's what most life subsists on deep sea, except in a handful of ecosystems like hydrothermal vents and methane seeps. And at those animals there, instead of eating these material that has come from the sea surface, they actually use the chemicals coming from the sea floor because of a symbiotic relationship that they have with bacteria that use those chemicals as a form of energy, and it's called chemosynthesis. And so we know that we have these methane seeps in Trinidad and Tobago's waters. We're really hoping, we're going to do our best to try to find some new ones especially. And yeah, we're going to do our best to find those. And so that really is, there are clues that we can use to find these seeps. And that's what we're going to be looking for. Rachel, you finished or not yet? Just tell me when you're done. So because of that Barbados accretion prism, we have more seeps than perhaps a lot of other places in the world do have. It's a place where seeps and mud volcanoes are found very, very frequently. We're also going to be investigating mesophotic coral reefs on a few dives. Those are later dives where we're closer to the island and we're on slopes. And then there's a range of other deep sea environments we're going to see. Here we can see a shrimp crossing our pathway. We're also going to be looking at the midwater to see what lives in the water column. And then we hope, as I've mentioned already, that we see some high density, and a hermit crab, lovely. This is the thing, this is so, this is so typical of the deep sea. You know, at a glance, you might think there's not a lot there, but when you zoom in, right here in this image, we can see a hermit crab. We had a shrimp swimming by, and right next to the hermit crab, there's a tiny little white branched organism, maybe one or two of them. Those look like bryozoans. And this is what the deep sea can be like. It can be like these small animals that you really have to zoom in for, but they're there. Or there can be absolutely incredible, and we do hope we see this, you know, deep sea coral and sponge gardens, right? Where just there's animals after animals. Wow, the crab is eating the bryozoan, the hermit crab. Are we able to zoom in anymore? Okay, that's okay. So this hermit crab, the shrimp really wants to get on camera. But this hermit crab has just trundled across to this bryozoan and is now proceeding to eat it. Oh, what is that? Yeah. And this happens a lot in the deep sea where you're like, what is that? And I still don't know what that is. A ctenophore? Ctenophore? Yeah. Now we can see it. So here we have an amazing ctenophore. Let's get some good images. Oh, yeah. Get some good images of that. Moving very quickly. How much of that is current? That's current? Taking it? Or?
Yeah, current.
So you can see from the particles and the ctenophore that just went by, there is a bit of current going by. And we can see how much marine snow there is in the water here. There's a lot compared to a lot of other places in the deep ocean. And that is likely because of the other unique thing about Trinidad and Tobago. Trinidad and Tobago sits in the Orinoco outflow. This is some sea cucumber poop that we're looking at right now. But so Trinidad and Tobago sits in the Orinoco outflow. So the Orinoco River is in South America and Venezuela, to be exact, I think. And that outflow is bringing with it lots and lots of nutrients. And so we expect that Trinidad and Tobago's waters are going to be very productive. And as a result, we're going to see some really, really, really cool things down in the deep sea, because that productivity, even though it's on the surface, also carries all the way down to the deep sea often. And again, you never know with the deep sea, but this is what we're here to find out.
Got a big clump of the sargassum coming by right now, actually up top.
And so what we're looking at right now, that's sargassum. We, in the Caribbean, we know this is that's okay. We can go to check out the other one. So sargassum is a floating algae. And it naturally occurs, except because of various global cycles being changed because of human activities. It means that sargassum has really started to bloom. And so we're getting these enormous sargassum blooms happening in the Atlantic Ocean that then carry over to the Caribbean. And we see them often in the media, coating beaches in islands where they rely on tourism, et cetera, et cetera. And so we see what ends up on beaches, but actually a lot of the sargassum ends up down in the deep sea. And so perhaps it's forming, again, as we said, a source of food for many deep sea animals. And that source of food does appear to be increasing given the amount of sargassum we're seeing entering the Caribbean. And again, these are the kinds of questions that we're going to be seeking to answer while we're at sea. Some what? Carbonate. Those things? Isn't that just where the ROV sat down?
Is it a no?
Quick question. All of this stuff here, is that carbonate or is that from where the ROV sat down?
This little
This
thing
is
in here,
the white rocky thing. Yes.
that's where we stamped the seabed with Excalibur.
Yeah, I think it just is bits of sediment.
I could look
Can we
at it.
pan over? Yeah.
Yeah. Right.
Oh no, Anna's on the.
What's wrong?
No. Is it? No, that's just sediment. Very muddy sediment.
It's very muddy.
Yeah, really muddy, right? So this is, we think sediment is more, but sediment tells us a lot. We can see that this is very, very muddy sediment here. Looks almost clay in some ways.
Yeah.
All the
How you doing?
sargassum out the waters.
Oh yeah, wow. There's a very large sargassum bloom just off where the ROV is being deployed. If you have that on your stream right now, I don't know.
Yeah.
But
Oh wow. We fired the heat pulse.
so if you want to learn more about the expedition that's happening now, please do go to the Schmidt Ocean Institute's website, schmidtocean.org, and you can find we have a whole expedition page. You can see, I think Alex has just put the sargassum up onto the live stream so that you can see what it looks like at the sea surface. This ends up on beaches. In Trinidad and Tobago, we have one of the highest densities of leatherback turtles nesting on our beaches. And this sargassum is now getting in the way of that on some of the beaches. And we can also see it's ending up in the deep sea. And we're not sure what that means for the deep ocean. So in about 3 more minutes, we're going to head off. And that would be great timing. And I'm going to hand over then to the next watch lead. We're going to be cycling through. You're going to hear a new person every hour or so is the plan. And help guide you, please do go and look at the cruise website. Our cruise is called Deep Wonders of Trinidad and Tobago on the Schmidt Ocean Institute website. And you can also read a little bit about each team member there. Please also, we do have a lot of socials happening where you can catch up on the behind the scenes, what's going on. We have a WhatsApp channel, which we'll be posting to social. We also have Schmidt Ocean Institute is, of course, reposting all the scientists on board. We also have Species, the lead organization. They are posting quite a bit. Oh, that looks like a lovely sea urchin in the distance. They're posting quite a bit, and they're going to be reposting on scientists so that you can see who's on board and follow along as well. While we're on board, we're not just using the ROV, even though I know that's what you were all here to see. We're also doing a series of different plankton tows. We are conducting CTD casts, so that is where we send down a water sampler that is able to measure different environmental conditions about the seawater and collect water samples into the deep sea. We are also... What else are we doing? Plankton, CTD.
Cores, push cores.
We're also going to be on some of the ROV dives collecting sediment cores so that we can look at what animals are actually living in the sediment. And excitingly, we have an experimental piece of deep sea equipment with us on board called Doris, the best name. This has been created by Ocean Discovery League and Blue Robotics. Doris stands for Deep Ocean Research and Imaging System. And this is an untethered underwater camera and sensor system that we are going to be able to deploy. We'll go down, collect images of the seafloor for us, and then come back to the surface, hopefully, so that we can retrieve it and deploy it again. And what is so special about Doris is that while this is a prototype we are testing, this new piece of equipment is really about expanding access to deep sea research because it is going to be very cost effective and easy to use. So this will, while we love platforms like Schmidt Ocean Institute's research vessel Falkor 2, we also recognize there are very few research vessels like that. So Ocean Discovery League and Blue Robotics are trying to create other more affordable ways so that more people can begin to explore and study the deep ocean.
We're all good, Diva.
Great. So we have finished our heat flow test. Pilots, we are good to set off. I'm going to hand you over to our next watch lead, Anna Metaxas. And please do follow along and please do share with family and friends. Thanks very much. So all I did was that.
I know. Yeah, they don't do much. The probe fully retracted. Starboard ramp coming in. Port's coming in.
Oh, I took a pen? Sorry.
That's what happens.
Who's our logger now?
Great. We power down.
Just now. Is that?
Yep.
Can we power down the probe?
Sure.
Are you sitting there? Okay.
probe is off.
Probe is on. Probe is on. Let me know when we're ready to move.
Okay, we're just gonna go to the south, right?
Okay.
Good plan.
So we're on. Okay. Good morning, everybody. My name is Anna Metaxas. I live and work in Nova Scotia on the east coast of Canada in a very, very much colder environment, although the deep sea there is probably about the same temperature as the deep sea here, around 4 degrees Celsius. But I'm very excited to be diving in the deep waters of Trinidad and Tobago. Never been down here, never seen this part of the world. Nobody has really. And so I'm very excited and very honored to be part of this first expedition and of many, hopefully, in this part of the world. So the way this dives work, at least initially, is because we've never seen here before, is very much like exploration. So we're gonna the ROV is going to drive along a track that we have predetermined, and we're going to be doing this in a very specific way so that we can actually be able to then take specific measures of who's here, who lives here, how many of them are there, and try to describe those communities. And so we will do those. We also collect some animals along the way because we will need to do some identifications to see if we have new species or new populations of species that have not been recorded before, et cetera, et cetera. So on that track, we're going to start sort of going uphill a little bit. We're now at about 1,044 meters depth. And yeah, we will sort of move forward. And we need to set certain parameters. Here's that urchin again that Diva was mentioning earlier, and you can see it has all those little bobs that stick out from it. Those are actually extensions of tube feet that you will see on any regular urchin that you see in shallow water. But in some species in the deep, these are actually toxic. And so if you touch it, you actually get a rash or even more. And so the other interesting thing that you see here is a lot of the animals we'll see will be red. And that is because, and those of you who are divers will know this, as we go to deeper water, we start losing color. And so a lot of the animals are red because red penetrates all the way down there. So off we go. And so we would like to do some uninterrupted transects of about 500 meters. So we would like to, we will be using, I guess, the side camera. Okay. We need to be no more than 2 meters above the seabed. We need to have a clear view. We want the camera as low as possible, but okay, with no obstruction, so no arms, no something in the corner there.
Yeah, pretty close, so if I look down anymore,
yeah,
you got that.
I know, exactly. So we just want to, perfect. Just a little tips. Okay, perfect. Right, and so now we'll move slowly along our transects and we'll see what we see.
Okay.
Thank you. Now, you may think there's an anemone there, that we're not seeing anything, that nothing lives here. But that, in fact, is inaccurate because there's a lot of animals that live in that sediment that you see. And they're small. They're not as photogenic as, the big fish that we see, or even sea urchin, sea cucumber, squid, all those animals that we already saw. They're not as photogenic, but they do a lot of important jobs. And one of the important jobs that they do is a lot of the carbon that rains down from shallow water hits the sediment at depth, and then these animals use it to feed, but also basically they sort of form this part of this cycle of carbon. So they process some of it, they eat it, some of it gets excreted, some of it gets buried further in the sediment, but some of them gets released back into the water. And what this contributes to is this global carbon cycle, which is how our climate is regulated. So you wouldn't even think that what is sitting down there in that sediment, that you think there's nothing there, but there is, because you can see those holes in the sediment, and those holes are made by, this animals that live in the sediment. And so that community that is under there actually contributes significantly to our climate. Who would have thought? So
A lot of fish down here.
a lot more
Yeah, we're
fish.
already
Yes.
seeing like 5, 6 fish.
Yes, and we're starting with, so there's a brittle star we're passing over, more shrimp. Just trying to check our altitude. One, yeah, perfect. So the reason we try to sort of keep a constant speed and a constant altitude above the seabed, which right now is about a meter above the seabed, is because when we then analyze this video, it's a lot easier for us. We know what area we're covering because we know how high we're above the bottom. And that way we can say how big the area is that we're seeing in the video. And so we can do some, more sort of quantitative measures of who lives here. Now, as we go along, we'll do those transects, but then we'll also stop at different points along the way to collect animals that we see that may be of interest. So, again, we're seeing a little bit of sargassum pieces here and there. Sargassum is an important, can be, oh, there comes the shrimp. Different one than the one we saw before, I'm sure. And there's one sitting on the seabed as well. Several, actually. See another one. So the reason sargassum is important, it plays an important role here, is that as Diva was saying, there's no light that penetrates the deep sea, and it doesn't reach that far. So there's no plants that grow. So animals have to eat something, and what they eat is the carbon that rains from above. And sargassum, in sort of regular amounts, provides an important source of carbon. But when there is too much sargassum, what happens is you get a lot of carbon arriving at the same time, and because there's not a lot of animals to consume that carbon, then the seabed can become anoxic. So low oxygen content. So what we're seeing going by now is a sea pen. So these are a type of coral that have a foot that's stuck in the sediment, and then the rest of the animal is up in the water and captures food. You may also be seeing these little tracks along the sediment. These are all made by animals as they move along. And if you have an area like this, which is muddy, that means that there's not a lot of current. And so those tracks will remain on the seabed for a long time. If you're in an area that, there's a lot of current, then the sediment will be much more sandy, and those tracks will not be as visible. So again, these are, although we can't see through the sediment, those indicators, if you wish, at the surface, like holes that we see as we move along, those tracks tell us that there are animals below the surface. And then, of course, in order for us to collect them, we'll be doing cores, as Diva mentioned later in tomorrow's dive, maybe. Anybody can tell what this white thing that comes by is? Hard to tell. again, we can see scattered pieces of sargassum. But for those of you who were on earlier, you saw that there is a big sheet, a carpet of sargassum that's floating at the surface above us. And so, interestingly, as Diva was suggesting, this is seaweed. It lives at the surface. Obviously, it needs sun to photosynthesize, like any plant. And the way it remains at the surface is it has these bubbles that keep it floating. So what happens is if there is a storm, and that seaweed gets sort of mixed down with big waves, it will because of the increased pressure, the bubbles will collapse, and so then the seaweed will sink to the bottom. And so that's why, although we have all that seaweed up there, you don't see carpets of seaweed down here, because I guess the weather has been pretty calm. But perhaps after storms or during a stormy period, you'll see a lot more seaweed arrive at the same time on the seabed. The shrimp over to our left. So again, you can see all the tracks here. These indicate that there's somebody under there who's made those tracks, but you can also see these little mounds. That also is an indicator it's a different animal that makes those mounds. And there are people that are actually experts in what type of animal makes what kind of feature. We call, that sort of, because they perturb the sediment, it's called bioturbation. And so they're experts in linking whatever the structure is to particular organisms, so that they don't always have to dig into the sediment to get the core out. Here's another coral coming through, another probably sea pan, and a shrimp next to it, sitting on the seabed. Another of those fishes. And so you can see on the seaweed here, on the sargassum, you can see a lot of, white bits. So what will happen to that sargassum is it will either be covered, and I was looking at some piece earlier that had this lacy covers organism, that's a bryozoan. And so that's an animal that lives at the surface and comes down with the seaweed. But also it can be colonized by animals along the way or actually on the seabed. So there could be little snails that live on it. Again, it's a source of carbon and food, and so they can sort of attach onto it and feed while the thing is rolling around on the seabed. So it does provide an important source of food, but it can be too much of a good thing. So. How far have we gone? Huh?
$70.
70 meters.
Where do you want to go?
Oh, we'll go about 500, but I don't know if you want to do it in 100-meter chunks, or what do you want to do?
Yeah, because she knows the time.
Here we're seeing again some sea cucumber casts. These are poop of sea cucumbers. sea cucumbers are amazing animals. They're Sea cucumbers are amazing animals because they just move along and they process all those sediments. So what that means is they ingest it from their mouths and then they poop it out in these structures that you see here in these casts. And so they can sort of cover large areas, and what they do is they remove whatever good nutrition is in the sediment. They process it at sort of as the sediment goes through their guts. They have bacteria that are in the guts that are basically digesting that stuff, and what comes out is clean. And so again, we're talking about the carbon cycle and climate, and so these are processes that are really important for that, because those animals sort of remove all that carbon from the sediment. And then, of course, when they die, the sediment the carbon gets released back into its cycle and continues the climate regulation.
So we said we're doing an hour. Yeah. Well, that was.
So that's about, it will be about 500 meters. That's what we want. Yep. Yeah.
Yeah.
Sounds good.
If I see something cool,
I'll be done now while we're doing.
go straight.
What's that? No, we're going straight. We're not.
Just go straight.
Yeah.
While we're doing the transects, we can, if you see anything exciting, just drop a marker and then we can always
Yeah.
use that.
Okay. Here we go. Something exciting.
What is it? Big stock sponge?
What is this,
Shirley?
Shirley?
Stalked glass sponge.
So we have our sponge expert on board, one of the top sponge experts in the world, a legend really, Dr. Shirley Pomponi, and she identified the sponge as a stalked glass sponge. So again, we're very fortunate to be able to bring our expertise here to this mission, because we haven't. It's all new for us, and so it's very exciting for us to be here to be able to share some of that.
You want a marker there or no?
No,
Yeah,
we're not
it's
going to
in
pick
your
up the one
WhatsApp.
sponge. Are we?
In the dive plan.
So sometimes they want a marker. So right here, that marker.
Yeah.
You can also get to it from the Google Drive. Okay, but if you do, then go save it out in the tech room.
So again, I think it's really important for those of you not familiar with the deep sea that are coming along for the first time for an exploration. Although this may seem like it's dead, it's really far from dead. There's a lot of animals that live in the sediment, and they do a lot. They have a lot of important jobs that they do for us, and obviously for the planet. So. For the longest time, it was thought that the deep sea was dead. Nothing lived there. Until the late 1800s, mid-late 1800s, where there was an expedition run by the global north at the time, that, however, circumnavigated the world and collected a lot of different samples, and basically proved that the deep sea is far from dead, that a lot of animals live down here. It looks like a sea pen to me.
Yeah, we've seen quite a few of those.
Yeah.
Ooh, what's this coming up?
Oh yeah, here we go. So that's some sort of octocoral, maybe with a brittle star on it.
So what we're seeing there are the stalks of those glass sponges.
Is that a sponge? What is that an anemone?
And they're covered
Oh,
with
anemone.
epibiots, zoanthids.
That looks like an anemone, right? Or a rock? What is that? Can't tell. Anyway, there's definitely an octocoral on there. And here's another of those sea pens. So if you wanted to collect some, shall we drop a waypoint? Would you be interested in those sequence, Diva?
Yeah. I think
Okay.
that if we're collecting things that are characteristic, we've seen
Yeah,
quite a few
we've
of
seen
those.
quite a few. So maybe we can drop a waypoint here. And the reason we do that is because we don't want to interrupt our transect, but we can come back and collect that. And perhaps we can make a note on the log about the waypoint and why we're putting it in. Oh, you're gonna call, yeah. They call it seepage, that's fine.
SEAPEN. Wood or something.
Wood. Where?
Off to the right.
Oh. Yeah, can't tell. Something off to the left, which it looks like was another one of those sponges.
Yeah, but I'm okay for right now.
There's
Pretty
another
far
one,
from the boat.
those
Yeah, you're not
octocorals.
seeing it.
Is that another sponge over to the right?
Looks it, maybe.
Yeah.
Can I remove your computer?
Surely.
Can I
Do
remove
you
your
want,
computer?
how long do glass sponges live for?
Okay. In the middle of
Oh. Okay. Middle of. Here's a somewhat larger piece of sargassum, and you can see all those, both the white encrusting that has come from the surface, plus some smaller white dots that are probably snails feeding on it. Ooh. Is that another one of those
Yeah,
Paleodictan? Yeah. So there's the structures that you see, all those little dots that you just lost from the screen. That is unclear who actually makes those. I mean, it's been the stuff of stories for a long time in the deep sea, trying to figure out, trying to sample, and trying to understand who makes those structures that you see, and we still don't know. But they're all quite repetitive. Is that? It's a bit. It's some sort of. Looks like a sea star rather than a brittle star. Yeah, the distance between the two laser dots is 10 centimeters, and we can use this to, if we're moving along, here's for example an octocoral on the side here. If we wanted to stop and measure it, we would put them right on it and we'll be able to tell how big that octocoral is. Some more stock sponges, looks like. And so we can also use it to figure out how big this frame that you see here, if we take a picture, approximately how large it is, so we can get an idea of the area that we're actually sampling. So it gives us a scale.
You go through Safari? Yeah. Or Safari. Safari works. I don't know what's called. Looks
like this seaweed has some sort of hydrozoid on it. Right bit.
Anna, we're on Mac Zoom, right? We're on Zoom to the end of the basket.
Yes.
Yeah. Sorry. Oh, what's this thing
Is it a
moving? Oh, no, the
transect?
halo Zoom.
So I got the boat just moving south all the time. They're not following over right now. They're gonna stop to the channel, but I got enough to the side so that they
Oh, there's a sea star coming up.
This is, yeah, 1 meter and like auto altitude. Once you turn that off, then it'll reset to DVL.
Okay.
But this is a transect too, where they don't want to like you're off to look at stuff or stop or see anything. If anything cool happens, we just take a marker.
So sea stars are relatives of brittle stars, they're relatives of sea urchins and of sea cucumbers, of all things. They have different ways of feeding. Some of them actually are predators, so they will attack and kill a mussel or a snail and digest it. And the way they do this is they have a stomach that they sort of evert, so they sort of take the stomach outside their bodies and they encompass whatever that animal, the prey is that they want to consume, and then they digest it, and that's how they feed. But then some of them are also, here's another Venus flytrap anemone with a brittle star on it. But then others just sit in the sediment and sort of absorbed materials from the sediment, so they feed sediment like the sea cucumbers do. So they have different ways of feeding. Here's more of those Bioturbation holes.
Well, I think they're gonna go 500 meters, so that's what that green is there.
Oh, that's our. Okay, that's our 500. Right. You can see a lot more. We're seeing more fish here. More Bioturbation. Wow.
A lot of halosaurs.
Our depth doesn't seem to be changing much.
Yeah, is it? Anna, can you the map
Hmm?
put up so for the where the sub is relative to the two? Usually,
Yeah, I can work on that. If I can do that.
yeah, just for not for the stream, just for this.
Yeah, there.
No, but with the Baffy.
Oh, with the back. Yeah.
No, just take.
Yeah.
So here you can see there's some holes in the sediment and slightly discolored mud around it, the sediment. So there's white around that hole. So often what happens is an animal lives in that hole, and then there's different strategies that animals have. But for example, if it lives in there, it will feed in there, and then it needs to sort of get rid of all the waste, and so that gets sort of flung out onto the sides and outside that hole, so that you can see the color changed. Or there may be shell remnants or whatever they've been feeding on that basically keep their holes clean. There's another sea pan. It seemed like we were oh, there's a teeny little brittle star on something that we're going right over. So in order for animals to live at these depths, they have to deal with a lot of pressure. Again, those of you who are divers, what happens when you go down 10 meters or 20 meters depth? So imagine going down 1,000 meters depth. Here comes a Brizingit. And so animals like fish have to have special adaptations to be able to live that, because fish have air sacs, and you can't have an air sac at that depth. You want to drop a waypoint for the stock? Yeah, let's drop a waypoint for stock crinoid, please. Stock sponge,
And also the sea star.
which was close. Yeah,
What do you want to call it?
stock sponge plus sea star.
That is a stalk sponge with a fish.
Are we? Yeah, we seem to be doing a bit of a dipsy doodle. Is the ship?
No.
It's okay? Okay.
I would go for you guys to see that spot.
So we don't want to do that. I mean, I know it's easy to get excited, but we're just trying to keep it straight line. So don't let us distract you.
Keep it at 60.
That could be. Oh, this fish. We see a lot of shrimp are sort of swimming by. Cascale, yep.
Actually, no.
Yep. It has a little barbules. And a sea urchin over to our right. Don't turn, just keep it on.
It'd be good to get one of those too.
The sea urchins, yes. Theoretically, we'll see more, like increase in density. There's another one on the left. Did you want to drop
Let's put mine.
oh,
Yeah, you can do it. Sea pan, whatever else, because that's common.
sea urchin. Stocked. Sponge. Ooh. That's a sea cucumber as opposed to a cast. And there's the cast. So you can see, if you look at this cast, you can see that there's a track that leaves from that cast and goes to the sort of bottom of the screen, the bottom left side of the screen, and that's sort of the sea cucumber as it's moving along. It's making those marks on the seabed, and so that's what we were talking about in terms of bioturbation. And because there's no current, not a lot of current, those tracks will remain there for some time. So we see a lot of, not a lot, but we see in some areas there is the seaweed actually either tumbles with the current, but if there's any sort of depression, any sort of hole, it will sort of pile up in there, and because it's concentrated, then often animals will, as they move around the sediment on the seabed, they will actually find it and stop and feed, and sometimes you can have aggregations of animals around those pockets of food. Again, because the deep sea doesn't get a lot of food naturally, because it doesn't have plants that grow there because of the lack of light, what happens is the animals are very attuned to picking up carbon sources of food, seaweed, or if there is in the spring, or there's a phytoplankton that sinks to the seabed. And in fact, they adjust their lifestyles to respond to those types of foods. So here's a sea cucumber coming up right under the sub ROV. And you can see the cast behind it, and you can see the path that the sea cucumber has taken. So you can see this path that is links to the actual poop. And there's further, some more further behind.
Busy morning.
It eats a lot, and you know, there's a lot of processing that has to happen. And then another one, definitely busy morning. Again, some sort of sea pen over to the, or octocoral over to the Dongo Strait. So although we see those interesting animals as we go, if they're off to the side, we try not to deviate from our transect, because then what we're doing is we're biasing to, counting whatever we like to see, as opposed to get a more objective view of the seabed, which is going along that straight line. So what we will do is we're dropping waypoints, and we'll come back when we're done with the transect. We'll come back and collect some of those animals, make some more observations of the behavior, and so on. But for now, we're sticking to our track. So here on the left side, you can see there's a hole, and it has multiple sort of tracks out of it. So that will be a worm, and a current worm. So what they do is they live in the hole, and they pop up, and they sort of lick the sediment. And so they do one lick, they retract, they do another one, and so it creates that sort of star pattern on the sediment. And I can't see pen? Can't tell what that is. So there's different ways that these animals will feed, even if they are in the sediment. So again, the reason we're not changing, so we have different tasks here on this particular dive. One is to get an objective, sort of representative picture of who lives on the seabed, and how many of them are there, how abundant they are. And so in order to do this, we have to keep a pretty steady track forward, because then we are at a particular altitude above the seabed, so we know how wide our field of view is, and we know how long it is. And so then we have this sort of strip of video that we can analyze using different methods, standard methods, to sort of count how many animals in this area. And for this reason, we don't stop, because if we stop, we're gonna mess up the sediment. We're gonna, it's soft mud, so it will be sort of resuspended. We may sort of resuspend animals that are in the sediment, etc. So what we do instead is we do this transect to get that idea, and then we're dropping waypoints along the way. So these are markers of where a particular interesting animal we saw. And so then the ROV can go back to that particular spot, and then we can look at the animal, make observations. So that will be the second part of this part of the dive. First one is we're on this transect, then we come back and we look at animals, observe their behaviors, and collect some of them for identification, for genetic analysis, and all those good things that will happen in the lab once we leave this place. So once again, although you probably think there's nothing going on there, I can see in the sediment there's so many tracks and there's so many holes that's actually suggests that there's quite a bit of activity below the sediment that we can't see. And in fact, deep sea sediments are considered some of the most biodiverse environments on the planet. So believe it or not, you don't think you're seeing anything here. And as my brother said at some point, this is so disappointing. There's nothing there. There's actually a lot there, and it's we just can't see it. Some we can. There's another of those sea stars coming up, so we may want to drop a marker here, please, for sea star. Oh, and a stalk sponge and a stalk coral there. Umbellula, yeah. So this is definitely a place we want to revisit. And there is something on the stalk of the sponge as well. A what?
We haven't seen this fish yet.
No, we haven't. Who knows their fishes? Grenadier, yeah, grenadier, yeah, macrurit. Nope, there's another Benedictine just left the screen. There's another those little red sea urchins. And a Benedictine. Now you should be able to see it. It's coming just about to leave the screen. Couple of sea pens, shrimp. Looks like another umbellula coming up on the right-hand side. Yep.
Squid.
Ooh. So you can see it, but everybody's excited in the room. There's another camera. This is the camera that is being streamed is the science camera, but there's also another camera that pilots use to drive with, and there was a big squid that went through there, but never made it all the way to the bottom. So apologies. Ooh, what have we got here? Shrimp. And another stock. Wow, that's a big one. Resting. So we have come up a little bit in depth, but not much, and it's a very sort of shallow increase, or not very steep increase. And so, because of course the seabed is pretty flat, that's why the sediment, and as we saw in the beginning, is really quite muddy. But um, as we see on land, as you get onto more steeper slopes, the sediment gets washed away, and so then you will start seeing coarser sediment, like possibly sand, and at some point, if it's a really steep slope, we will have exposed rock, and that's where we can start seeing things like, coral gardens, deep water coral gardens. Because they require hard rock to attach to, and so the only corals that we'll expect, coral type organisms as we expect to see here, would be sea pans, and we've been seeing quite a few of those because sea pans have a foot that it's basically in the sediment, and they're anchored with that foot in the sediment, and so they're the ones that can grow here sometimes, and we may see those sea pans can actually form big fields, but that again depends on a number of conditions, and we may or may not see any, but we're certainly seeing quite a few of them along our track here. There's a dig too. Is that a stalked anemone? Or Cereanthid? So the other interesting thing is anemones. Anemones are a very, very diverse group of organisms. Some of them are attached, they need rock, hard rock to attach to. And there's another group called Cereanthid anemones that are actually also live in soft sediment. And so they have this trunk that sinks into the sediment, and that's what anchors them into the sediment. And so anemones also feed on different particles, or they capture zooplankton. So again, they're very, very diverse. They're also very hard to identify. Again, because they're very diverse, they can be the same species, can have many, many different colors. And I suspect we're going to start seeing some just because they are quite abundant, and they do live in sediment. So I do think we saw one of those that passed by. I think that's another sea pan coming up. Eh. What's our temperature? 5.37. So animals, as many animals, migrate vertically during the day. So if you're a fish that uses eyesight to catch your food, you need light. And so animals that are eaten by fish tend to migrate to the deeper waters in the daytime so they cannot be seen. And they come up to the surface at night to feed themselves. There comes another sea pan on the side. And I don't know what that is. Oh, is that a... It's a leaf. Is it a leaf or is it a sea biscuit? It's a leaf. Yes, it's a leaf. We're going to start fantasizing soon. Right. So the animals vertically migrate, and so they go up at night to feed, and they come back down. And those migrations can be thousands of meters, right? So definitely there will be animals vertically migrate from here to the surface and back down. It's not inconceivable. Those distances are not inconceivable. Now, the temperature down here, so there's a question about whether, animals are sort of less active because it's cold down here. Temperature is cool. It's especially for Trinidad, it's below 5.5 degrees Celsius. So it's not even that they lay low in the daytime or at night because it is because it's colder. Metabolism slows down, and so animals may move slower because it's better to move slower because you save energy. The other reason that the animals are going to move slower is not only because of temperature, but also because they don't have a lot of food. And so you want to be as efficient as possible in terms of maximizing energy that you take from whatever food you receive, but also minimizing how much of that energy you use. And so, yes, animal will be slower, both because of temperature, but also because it's an adaptation because of the low amount of food that they get relative to what they would have if they lived at the surface. Oh, that's another sea urchin. So this sea urchin is interesting. It's coming to our right. It's possibly an Ariosoma. You see that it has the little bobules right on top of it. So these are actually full of poison. And so they will, if you touch those and you burst them, it's a defense for the urchin. So they're actually quite toxic, those particular sea urchins that can be quite toxic. And it's not just the prickly part, it's actually a toxin in those. Whereas there's another one coming up, which is a different urchin, this red one that you see here, and although I don't know, it doesn't appear to have those particular structures that are toxic. Okay, so we'll pass it on to our next hot seat person here. You've heard from Rachel before, so she's coming back and she'll take you on the next part of this trip.
All right. Hello again, everybody. This is Rachel Lauer back from the University of Calgary. And unfortunately, I'm not a biologist. So, I mean, I know enough to be dangerous. Sometimes I can tell a rock apart from a sponge, but like that might be a sea cucumber. Yes. Okay. Yep.
First test.
Yes.
And look at all the pool along the way.
Yeah. And yes, is that from the same sea cucumber? Okay. Another sea cucumber. So I'm here with postdoc Rob Perrin, and we are conducting heat flow measurements and thinking about the hydrogeology of this system. Is that another one of those?
It's a stalk of a dead hyalonema. Looks like it's got an anemone attached to
Okay,
the
someone
top
just
of it.
said so many
And so we have to
syllables
along the
that,
stalk.
yes, it's past now. Yep,
Are you in Sinaloa?
I know.
Put it in.
People forget I'm not a biologist.
Yes.
So this is a really crazy place to be as a geologist because we are in the middle of some of the most complex fault systems that I've ever worked around. We have a subduction zone to the northeast of Trinidad and Tobago. So we have the southern part of the Atlantic plate subducting under the Caribbean plate. And while it's doing that, it is scraping off tons of sediment. And where we are right now, there is up to 10 kilometers of sediment that has been scraped off and piled up. And that's where we are right now. Also, just the northern portion of Trinidad is actually forming the plate boundary between the South American plate and the Caribbean plate, and that is a strike slip fault. So this area experiences frequent earthquakes, and as everyone knows who lives here, there's also a lot of mud volcanoes, both onshore and offshore.
Did you hear about the beach seagrass after the Venezuelan earthquakes?
Oh, yeah.
The beach went up like 20 feet. up.
In Trinidad.
Yeah.
Yep, I read the article. Yeah, so although, oh, glass sponge. Is that right? Is that a glass sponge? Oh, man. I'm getting better at this. Yeah, so I'm going to stick with my expertise and talk a little bit about the local geology. Obviously, we're quite a ways from where the earthquake happened last week. But even this far away, you can have some deformation and motion in response to such a large earthquake. And there's some local articles that have come out suggesting that in response to those two events, actually, it's a doublet. So that means there were two similar magnitude earthquakes within 1 minute of each other. The first was a 7.2 and the second was a 7.5. And when you have that much motion that close to the surface, there's a lot of energy, and that can cause places as far away as Trinidad and Tobago to move a little bit in response. Certainly, we didn't experience anything close to what happened there. So that scraping off of lots of sediment that piles up is this process called subduction. And those types of, here we have multiple plate boundaries, as I said. We have subduction plate boundary where one plate is going under the other plate, and then we have plates sliding past one another like what we call a strike slip fault. So the mud volcanoes that are all over the place offshore, and I think there's more than like 20, or I actually should know this, but at least 20 in the southern portion of Trinidad. And these are not volcanoes in the sense they're not spewing lava or molten rock, but as the name suggests, they can occasionally erupt mud, which is the result of really high fluid pressures. And the way you get really high fluid pressures is in places where, there's a little shrimp. All right, these are too easy, even I'm getting them. And when you have places where a lot of compression is happening, where you have two plates grinding past each other, or one plate going under another, what happens is whatever is being carried to greater and greater depths gets pressurized. And when that fluid pressure increases enough, what is sediment can actually start acting more like a liquid and become buoyant and try it and make its way along those faults to either the land surface, where they form these mud volcanoes that I've seen pictures of people actually getting into them and swimming around in them, or offshore, and we will likely see some of these on this trip, where they form submarine mud volcanoes. Despite the fact that they're not as dangerous in the sense of lava and the hazards associated with that, you can have a large eruption that can bury a large area if you have enough fluidized mud. So because the same processes also tend to form oil and gas provinces, they tend to occur in places like this. Okay, what's that guy? Or gal? What's... Anyone? Biologist?
Sorry, it's a fish.
I know it's a fish, but yeah. No? No one's gonna help me?
It's a big fish.
It's a big fish. Okay. We have a sponge expert who says it's a big fish. Yeah, so in addition to water that is part of what gets spewed at the surface of these mud volcanoes, there also are gases like methane and CO2. And I don't have a specific count or like a census of how many of them there are offshore here, but that's one of the things we're doing is mapping the seafloor and trying to identify not only what's there as far as animals, but also what kind of structures. Sea star. Is that something? No? Yeah, so for the initial part of this dive, we are just observing what is present and trying to cover as much ground as we can prior to conducting any biological sampling or sampling of fluids, etc. There's a good question in the chat. I am curious to know the answer. I have, as I mentioned, read that there was some surface uplift along a fault in Trinidad in response to the earthquake. This was observed by someone who lives on the beach and noticed that what was previously underwater is now not underwater. So the estimate was there's up to 20 feet of uplift. Sorry for the units of feet, but some of us think in feet, some think in meters. But that sort of uplift is common in response to either local or remote earthquakes. Sort of an adjustment of the ground surface in response to all that stress release that happens during an earthquake. Hi, Jenny. Yeah, it's good to be back. The best ship. And there is a...
Is
that a brittle star?
Stalk.
And,
It's a dead stalk of a dead
You need to talk in less. Okay.
Okay, that's end of Transect.
Dead-
We had the brittle stalk.
Brittle star. Okay.
Yep.
Stalk of some
End of Transect.
dead dead sponge. Got it.
Right?
All right. So we just ended the transect.
Yeah.
Okay. And then
But it's very much alive. It's got anemone-like things growing on it, so very much alive.
cool. Okay. There are living things. As far as the next transect, what's
So we're not doing, now we're gonna go back and collect some of those samples.
okay? So
We need
on
to
the
get back
same
to the
line.
Lloyd points. Rob is here about his sponges, so there were sponges, so remember we need to get water first.
Yeah. I'll just take 2 bottles of water and put them up there.
So you're gonna need to cover, so we have a bunch of
Bridge RV.
waypoints that are sponges.
All right. So we've just completed the biological
Bridge
transect.
RV.
That just means that this portion of the dive
Okay, Rafa, we're closing this Transect.
is done, and we're moving on to our next objectives. We have
keep that in mind, right?
multiple sponge experts here in the room, and it looks like we may be doing some sponge sampling along this transect.
Thank you.
So we need to go back to our waypoints.
But we don't know.
Yeah, we do, they're all marked. There's the bigger one behind us.
Alright.
That just came out.
Yeah.
One
So-
more likely.
Huh?
Let's go.
So we need to go back to the waypoints.
Bridge RV.
One by one in reverse.
Okay.
Go ahead.
We've already
Okay,
done them
Rafa,
all.
you can start moving.
We haven't sampled.
Please.
No, no,
Go
I know.
back to
Copy.
Yep.
Okay.
So there's some sponges for this.
Robert.
So we need to go back to the waypoints. So let's go to the first one, which is a sea pen one, and then the second one is a sponge. And we'll see how far that is.
Okay.
But we don't have to go, we can go fast as they're going. We don't have
Yep.
to go slowly and then get and then drop.
Okay. See, we did drop waypoints where we want to sample. Okay.
And they have them all listed there and that just to see that gradient.
Yep.
Zero waypoints. Just getting straight back to the first one.
All right. So
Um,
he's heading back now.
So we need to get there. And then when we get there, I guess. Yeah, I mean, Rob should be saying perfect. And that's not how this works. So basically, Rob will need to find Niskin,
Mm-hmm.
and then he needs to collect the sponge.
Yep.
Let me check.
All right.
He starts.
So based on the observations that were made along this transect, we've dropped some waypoints at locations where we'd like to do some sampling. And that sampling will include fluid. One of the researchers on the vessel is using eDNA, environmental DNA, and my understanding is that the fluids that have interacted with and potentially have pieces of animal DNA, that DNA can be identified through those fluids. So we collect those fluid samples on the sub by firing a Niskin bottle, which basically slurps up some water that can be analyzed here on the ship. I don't want to talk too much about what Robert's doing because he's going to talk about it later today. But a little preview is it's very, very cool what he's doing. And it's basically, he has an entire lab on the ship that is the size of like 2 iPhones. So he'll tell you more about that.
I don't know if you could see it on your screen, heading back to the first waypoint called C10. So I'm just trucking along. So you can speed up as you need to, right? Thank you.
Yes, we're going a little bit faster for this part because we know where we're going and we have already seen this, so that we can cover as much ground as possible over the course of this dive. All right, we're coming up on a glass sponge, I think.
Beautiful. Hyalonema is its name.
I'm not going to say the. Okay.
What's a hyalonema?
You have to turn back to airport 2.2, turn clockwise. So coming down and turn from the clock to the right side turn. So you get up and the turn clockwise.
So, can somebody tell me where?
We're heading to the marker up there, or C-pen. Okay. That's the closest one. That's
Closest sample?
yeah, okay. So we're just trucking.
Got it. Cool.
change the plan. So we're not going to go back to this time. We're not going to go back to the waypoint. So if they're trying to go back to the waypoint,
They are.
stop them.
Okay, that's what they're doing.
Stop them.
Okay, guys. Apparently, there's a new plan.
Okay.
I've just been informed. What is the new plan?
Okay, so we're not going, so we can turn around from where we are and we can just go along that transect, go past where we stop and keep going here. So the idea was to go back all the way back in. But, yeah,
So you just want us to turn clockwise and head back
yeah,
south?
right. And then now, but we're going to be.
I mean, it looks like south there, but well, it's not. We're not heading south, are we?
At the moment, we're heading back north.
So we need this information. This is the problem.
Yeah.
We need the map.
Hey, Jules, could you put their map up on there?
I can put it up.
This is the problem.
The link. Okay, the map right now is down. She's working on it. So as soon as she get it back up, she'll put it there.
Zoom out on your end.
Uh-huh.
Okay. That's what they're saying.
We stopped and when I came on, we were just heading south. So I just took over from Zach. I was waiting like one there to stop.
And
it.
where's waypoint 2?
That is. So we were heading south.
So we dropped at the north. So we're going down, yeah. So this is not much of it. This is the double the hill.
We need to go in the plan.
I know, that's where we stop it. So we have to get them out closer. So we're in the camp.
We're not. Well, zoom out.
Isn't this the camp? We're in it. We should be here.
This is not our bathymetry that we. Can we zoom out?
We have this.
Can we zoom out on that map? One that's up. This one. Yeah.
So we're there, and we're going there. We
It's-
wanted to be here.
Yep.
So we're just heading
Is the yellow line on here where we are, or no?
I don't think we're there.
Okay.
So that's what happened, right? It's that sort of the smile.
That line is not where we
No,
are.
this is not. This is the center of
This is where the
the
camera
center.
I don't know where we are.
Yeah, we aren't getting a location right now.
I think we're there. Yeah, I know.
Oh, no.
But it's... So,
All right, so we
So
just
there's no-
continue towards waypoint 2.
Waypoint 2,
Yeah.
head south.
Yep.
should we move to the...
We don't know where we are.
So that, we can superimpose that. Do we have our position relative to this?
We don't.
No, it's not working.
It's not working.
They're trying to fix it.
There's no battery on the other.
There's no one.
Battery on the ship's mapping.
All right, so it is currently lunchtime, so you're going to be hearing folks come in and out and all that. And also, the scientists right now are sort of figuring out where we're wanting to go on this dive. We're about 2.5 hours in on the stream here. This is intended to be a 12-hour dive, I do believe.
right, so somewhere to the east or west of us is a 30-meter canyon. So that's what we were trying to find on this map. Yeah.
It's probably like here.
Yep.
This is
So
what
it's
we're looking
going
for.
to be to our east.
It is to our east. Okay. So then, if we could head towards that, and then we can follow that.
All right, so if you didn't catch that, there is a something of a canyon feature here near us, and we're just going to try and make our way over to it with the ROV.
Yeah, so sorry for the hiatus there. We were consulting 3 different maps and trying to put them together to figure out exactly where something we mapped overnight. So in this area, there's what is a submarine canyon with about 30 meters of relief. So between both sides and the canyons, about a 30-meter difference. These canyons tend to be places where you would expect methane seeps and other rocky surfaces that might provide potential habitat for the biology that we're most interested in. And as you might imagine, it's really difficult to locate yourself on a moving ship, in a moving sub. I think it's called a 3-body problem, trying to locate everything precisely. So ideally, we're heading towards that canyon, and we'll continue heading south once we get to it.
You with Point Bridge.
So because we haven't actually been to this part of the seafloor with a submersible or humans, and we haven't really mapped in high resolution any of what's offshore, Trinidad and Tobago, what we do overnight is use the ship's multi-beam, which is basically using sound to map the seafloor and all the features on the seafloor. And that's basically the topography of the seafloor. So the little hills and valleys, once those are mapped, we try and make our best guess about places where we would expect biodiversity. A lot of the stuff that we're interested in is like corals and other creatures that can't move around are going to be found on rocky outcrops or other places that allow them to thrive in this really dark, cold environment. And I think Anna talked previously about chemosynthesis and the fact that these vents, oh no, that was Diva. These vents can host specific bacteria that can take chemicals and turn them into energy. Do we want the sponge? Okay.
Along the way, so we decided to do it this way. Just
Okay.
once we've done it, so
Okay.
we can stop and get our change or whatever.
Okay. Somebody needs to tell me if they want, like, the person that wants it. Yeah. Okay. All right, we have another fish hanging out, giving us the side eye. Okay. All right. Yes. C pen? We're gonna get it? Okay, we would like to sample this C pen. Okay, so now that we've finished our transect where we're just observing and cataloging and taking inventory of what we see, there are lots of biologists on the ship who have different expertise, and part of this is conducting biological sampling. So we're going to sample the sea pen that was just to the right of our view. Yep. 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 Uh-oh. So there was a C pen over to the right. I think we've rotated away from it, maybe? You know where it went? Should we keep going? All right, let's keep going. Head towards the... I don't want to call it a gorge because that implies it's large, but a canyon, that's what we're calling it. Oh. Is that an urchin or seaweed? This is why you don't put me on the hot seat. It's seaweed. Don't collect that. It's a tumbleweed. Seafloor tumbleweed. Oh, and another shrimp. Yes, sargassum tumbleweed, exactly. Pew, pew. All right, so if you're just joining us, we are, I'm Rachel Lauer from the University of Calgary. I'm not a biologist. That said, I am hoping to be one in my next life, so I'm learning a lot while I work with biologists. And we are offshore Trinidad and Tobago. This part of the seafloor is the result of lots of complex deformation between tectonic plates in this region. We are on top of what's called an accretionary prism, where sediment from the Atlantic plate is being scraped off onto the Caribbean plate and forming this 10-kilometer thick, massive pile of sediment. There's also, on the northern part of Trinidad, a strike slip fault that forms the boundary between the Caribbean plate And, oh, what is this? Oh, another sea cucumber. Okay, can we hold up and the procedure for this, as I've witnessed others do, is to, prior to putting it in the bio box, we're going to do some glamour shots so we know what it looks like before it went in the box. And some close-ups of it while it's in its natural habitat. Cool.
Great, you could come to us.
Probably Bridge, you're not very clear, sorry.
Yeah, you could come to us though.
Where does that?
All right, so we're slowing down to first get a, prior to sampling, getting a close-up view of the sea cucumber. This is, I believe, the probably 8th or 9th of these that we've seen today. All right, and these lasers, we use these because they are calibrated and at 10 centimeters, so regardless of the distance away from the sub, they allow us sort of a scale bar for what we're looking at. Because obviously it's really difficult to determine size and scale when you're looking through water and zooming in and out. All right. Okay. I think we just, yeah, I think once we, okay, so we don't have, so I think this can, I mean, I know we're a ways away from sampling, but everything's open, so it's not huge, I don't think. So it can probably go in its own bio box.
Which bio box you want?
Whichever, this one, is that the one? Yep. Yeah, I don't know if anybody saw earlier, but before we did the heat flow probe measurement, we used a stick to sort of determine how soft the sediment is, and it's really soft. Just the weight of the stick in water, which is actually quite, quite light. It was actually sinking into the muds. So we're in an area where what you see as mud is actually like soft serve ice cream. It's super porous and mostly water. So porosity is a measure of how much sediment there is relative to space between it. And in places like this, it's just a lot of water. we're talking about like 70% porosity. So only 30% of that mud is sediment and the rest of it is water. So anytime you get close to it, going to kick up a little cloud, which probably for an animal living down here looks like a massive sandstorm or something.
Right?
Yeah.
Or both?
Or both.
Or isn't that a sea star also?
Now,
All the things?
all the things are appearing in front of us. There was a sea cucumber, but there's a star and an urchin. Would we rather have those, or? We zoomed in and got the glamour shot in situ. Yeah.
Got the cucumber and
Okay.
then we can always think about
Okay.
it. Unless it's really, really hard.
Yep. Wait, what's my X? Those are our skid marks, I think.
Yeah, I know. But where is this?
But I think he's right
Oh,
there.
yeah.
He's between. Yeah, you can see the clays actually. They said they were gonna put it in.
Yeah, and then priority will be
Yep. Got it.
after.
Put it in here. Well, not. Yeah.
Let's see.
We'll see where it ends up.
Oh, that's a beautiful sea cucumber.
All right. Wonder what that hole is next to it.
It's on lock now. Once this comes on, it's gonna pick it up. Just like this.
so that's definitely not how you spell sargasso.
Just for that. This one? That's what you want messing up to. So you do.
At least it's blowing away pretty quickly.
Yeah. I think so this more this way.
We all have the plan.
No way.
Are you trying to go here?
We were in the bars.
Even on the flat?
No,
No.
we want to be in the canyon.
Or where do you mean on the map?
Well, I don't know where we are.
This is where we go. Right us?
No.
Oh. I'm not too sure.
Trust?
Yes.
Let them know. So see, it keeps it coming. And if you just, it's moving. Once you make up a mark, it's going to override the lock. So you not sure if it's still there.
I think it is.
It's there, sir?
Yep.
Where?
It's just a little dusty.
Can reach under the desk. There are plenty of outlets.
Yeah, this is really fine-grained sediment. And we would like to, while holding it up before it goes in the bio box, get a zoom in as well. You want them to turn the arm?
If we can.
If you
yeah,
can, just
and
to,
we
or not the
also-
arm, the hand.
We did
I-
lasers on
not-
before.
We did lasers on in
Great.
situ.
Great.
Okay.
Yeah,
I
great.
think they're ready for the bio box.
They are bigger than the looks.
Yeah.
This is the Right, what's the one in it?
The bottom
My box 3.
3D.
Yes, previous trip we collected one was a purple one. Beautiful. Like massive.
No, the purple ones get like a baby.
Just this one.
D. 3D.
Is there anything in B?
Nothing in any of them.
One's watching a little.
Okay, so he's in there. It's in there. All right. So the reason we takes our time to video in situ with the lasers on prior to sampling is so we know what it looked like in its natural habitat prior to sampling. So this is actually our first sample of the cruise. Okay, so it's in BioBox 3D.
That's it for you? So you want to continue?
Yeah, do you want,
500 meters.
Diva, do we want, there's also urchin.
Straighten up this
Do
cover.
we want to keep heading east,
Before you left off.
which according to our calculations is where the canyon is?
You could see here.
Sure.
Yeah, okay.
When you left off.
We're saying
During this dive, do we want the next transects to be also 500?
I think we want to bring the flashlights up.
Yeah, right?
We can be 500.
Yeah. So,
Towards the canyon.
so I think we should place those waypoints from the start. No, from starting we place like 100 meters waypoint for them.
On the start. On the start of the transect, so the start of the dive.
The transect.
Yeah. Okay.
so you know when to flash.
That's it, but their position was the batch. That got broken in port, so I had to kind of redo some strings, but.
All right, so as I mentioned earlier, we are having just taken our first sample of the cruise, sea cucumber. We are heading to the east, where we should encounter a submarine canyon that we mapped last night, or a channel.
Yeah, that's great. But it will be working.
Great, you could continue with the move.
Señorita Luis.
Oh, good. Who's
Yeah,
that?
you could continue with that move.
Okay.
But, I mean,
Yeah, I wasn't sure. I was like, seems like.
Hello everyone, thank you for being here. My name is Jamie Lee Luchin and I am a graduate student at the University of the West Indies. And if you're now tuning in, we are exploring the deep sea of Trinidad and Tobago. This is actually only the third time scientifically that we are laying eyes on our deep sea. So it's very exciting and very new. Oh, a beautiful sea star. And there are lots of sargassum seaweed on the seafloor, as there are large mats floating above on the ocean. Some of it sinks down to the seafloor. Oh, fish. Beautiful fish. Oh, another sea cucumber. I'm actually one of many students from the University of West Indies here on this expedition. And our yes, another sponge. And the majority of our team is actually made up of Trinidad and Tobagonian scientists, which is very exciting. We've actually been seeing a lot of sponges this morning, which is quite exciting because historically we've mainly used trawl nets to explore our deep sea, which meant that a lot of the more fragile organisms like sponges, ctenophores, jellies, trawl nets were not designed to capture such soft-bodied organisms, and so they are missing from our records. We have a few, and they're missing not because they're rare, but because the methods that we've been using were not designed to capture them. So every time we see one, I'm getting very excited. That was a sea pen that we just passed. another fish. We've actually been seeing a lot of fish this morning. A lot of fish, a lot of sea stars, a lot of sea pens, a few sea cucumbers. And we completed our first transect this morning. And we collected our first sample, a sea cucumber. And if you're wondering how big some of the animals may be, the 2 green laser dots you're seeing on the screen, those are 10 centimeters apart. So that sort of gives us an idea of how big the things we're looking at are. You see the big sea star to the right of the screen. Ooh, what's that to the left? Is that a... Yeah, it's a ghost shark.
Yeah.
Ah, what a beautiful ghost shark. Ooh, and he has parasite on his head. He's a ghost shark. Yeah. We also saw a ghost shark back in 2014 when the EV Nautilus came to Trinidad and Tobago for 3 days. Beautiful. He got spooked.
Yeah. Very cool.
Another sea cucumber. We are currently diving in Trinidad and Tobago, but more specifically, we are closer to the western side of Tobago. And this is dive number 1 of a month-long expedition. Some more sargassum seaweed. Looks like there may be an urchin in the seaweed. Oh, it's sea sponge. Do we want to collect this? Do you want to collect the sponge?
Yes, sponge, okay.
Sponge?
Fire the, yeah,
Okay.
fire the Niskin.
Yeah.
One Niskin.
On the skin?
Before anything else.
Okay. So we want to fire in the skin and then collect that sponge. Yeah. So what we're actually looking at here is a glass sponge, and to the left of it is another sea cucumber. And so we are actually hoping to collect that sea sponge. It's called a glass sponge, but before we do so, we want to take a water sample.
Big little waters over the north area. Like you're not the one who's supposed to be quiet.
RV Bridge.
Yeah, Bridge. Yeah, Bridge, you could come down for us.
You're coming through really broken. Is the mic loose?
I'm not sure. You can come down for us.
Okay, coming to stop. That's a lot better as well. Cheers.
Roger. Niskin fire.
Number 2. Niskin fired number 2. Number 2. Yeah. One and two fired. Niskin number 1 and 2 fired. Yes. So now that we've collected our water sample, we are hoping to collect this glass sponge that you can see here. And there's lots going on. There's an urchin, there's fish, there's a sea cucumber.
And you want this in a box, right? Bio
Oh,
box.
it's just a-
Oh no. It's a bio box. It's a bio box. Fine.
Yes, yes.
And either one, any
I think
exactly
it would
what we
fit
do.
in a Falkor.
If you want peace.
Okay, no, we want the whole thing. we like the whole glass sponge.
You want the root of it.
Yes, everything. Yeah.
Good.
And there on the floors at the bottom of the sponge, we can also see a brittle star, I think, or it might be a sea star.
See that on the bottom? Those ones at the top.
Wow. Oh. If we can just, we can just have some angles of him if possible.
You want some?
Just, you know, just to look at him a little bit.
Some.
Cool. Wow. Oh, we can also see little arthropods in the sponge. Those arthropods.
Yep.
Yeah, yeah, little arthropods living on the sponge.
Yeah. What do you want? Okay.
It's like fiberglass, and it's okay if it breaks.
There we go.
Very nice.
Beautiful.
Nice.
Woo!
Nice.
And that is our second sample of the day. Good job.
Nicely done.
Yeah?
Slurping.
Slurp the urchin.
I think it's pretty small.
Okay.
Slurp.
We can slurp the urchin. We're also going to collect that sea urchin that we can see right there.
This is after after rotate. Just go to number 1. Let's flash it to 2. And then pick another one. Rotate to a different one for bridge.
Okay.
Huh? Just to get rid of anything that's already number 2.
Okay.
So if somebody needs the bridge on it, it's already.
Yeah.
So you always flash with that sound.
So earlier this year, a wonderful team and I published a paper on essentially everything that we know up to date on our deep sea. And we discovered that we have 451 species on record. And of those 451 species, we had some physical samples, but about 90% of those samples were sent abroad. And so the very exciting thing now is the majority of these samples collected here are going to remain within Trinidad and Tobago, either at the University of the West Indies or at the Institute of Marine Affairs. And so local scientists and local students and researchers will be able to benefit from the research that we're doing here today.
Density so it's too big.
Yeah, we can put over 6 to the bed.
Yeah, we just got close. It's too big.
Let's do it. So it's a much of
Yeah. Yeah, that won't fit in the slurp.
Let's work on that. we're changing just now. There's a
So now we're just zooming in so that we can have a close look at it before we sample him. See him in his natural environment. Now, as you zoom in, there are always other species you can see in the surrounding bed.
So my first trip here, I did the first 2 months for this, and then I'm going to for the third month. It's really incredible. It's amazing. I always busy working with oil and gas or shipwrecks and vessels. It's a different world, and it's nice because the things I can see now, I can ask for you guys. It's so nice.
Nice photos. Nice photos, too. Good.
Which the laser's on.
So we're able to size it.
Yeah, that's good. Yeah. Probably catching that will come up. Yep.
Oh, we think that's the picture.
Yeah. Great. Just on the left. Yeah. If you look at it, in fact, we can.
Well,
He might break. We're planning to put him in Biobox 2A. Perfect. Thank you. Oh, look, another shrimp.
Continue east.
Yep, let's continue east. Let's keep exploring.
Oh no. Well, that's going to be used for
Bridge, you could continue with that move.
Continue with the move.
Okay.
Yeah, right.
That one? No, I think that's one of these pubs.
On the sources. This essentially is a transect, but the music is okay.
Yeah.
But I don't know which one is not go up. That's the
You can come in.
good times.
Yeah.
Yeah. It is a star.
She's on it.
Oh, you want to collect it? Oh, I
Like
think
the
you want to
sea
collect
stuff?
it. Where on a? Yeah. But yeah, we just want to zoom in before we pick it up. So that and then we can do it like we said.
For anyone joining us, we are about a third the way through the planned time for this dive, assuming that holds. This is the first dive of this expedition, and you're watching live footage from remotely operated vehicle Sebastian, just over 1,000 meters deep.
Perfect. We probably turn off the lasers now.
Currently, we are looking for about they said 30 meter canyon that was a bit off to our east. So we're heading that way, see if we can encounter it.
For some reason, they deleted waypoint 1. Waypoint 1 was right here. So this is our map. So this is north, this is south. And this is the location of Sebastian. This is the location of Schmidt. Oh, there's waypoint 1. So waypoint 1 is there. There's waypoint 2 to the south. Here's all the markers to the transects. This is up here.
Nice zoom.
Yep. Yeah, this is the first dive. We're figuring stuff out. But if you're not sure where you are, you can just ask the co-pilot to.
Got a nice zoom here. Nice zoom of the sea star. See those tube feet moving slowly underneath. Yeah, the visibility's been kind of come and go. Really depends. There's something of a current down here. It's not out of control or anything, but it's not necessarily playing nice with us sometimes when we get a little cloud on set down. So we're going to try and collect this star now.
Yes, please. About wax.
Nice look at the texture. Sea star. Oh, it's slipping a little.
It's not loose enough.
Oh, okay.
Try and just flip it.
Yeah, I just tossed up a link to our vessel tracker there in the chat. And if you click that link, you'll be able to see both the location of Falkor 2, the research vessel, as well as the ROV Sebastian.
Okay, while we put that sea star in the bio box, I'm gonna hand it over to Charisse.
Nicely done.
Hi everyone, this is Charisse joining you for the next hour. So you guys would have just seen that they've collected a sample and that was a sea star and it was put in one of our bio boxes. This is my first expedition, so I'm happy to join you guys. This is an exciting time for me, an exciting time for you guys, and an exciting time for Trinidad and Tobago in general. Yeah, so like I was saying, this is an exciting time for Trinidad and Tobago. This is our first Trinidadian-led team of scientists. There's 25 of us on board, 16 of which are from Trinidad. And we are exploring a location that has not before been seen. We are currently west of Tobago. And we've done our video transects earlier, and now we're exploring and taking samples as we come across them. Currently we are at a depth of 1,023 meters, so pretty deep, guys. And we'll see what we see. So far from the feed, anyone who's there earlier, you would have seen some glass sponges, sea cucumbers, sea stars, urchins. There was a ghost shark earlier. Sorry to those who didn't get to see it. Hopefully we see another one. You'll see we're passing over a shrimp right now. Bright red coloration at the bottom of the screen. In the deep waters, you lose the color red first. So anyone who's ever dove, you would know this rule. But just so everyone else, as you increase depth, you lose the color red. So a lot of things in the deep sea, you will see have bright colorations. But the other animals are not seeing that.
bridging, continue that one.
Continue in the main.
Lovely little fish telling us hello. Hopefully he leads us to something else as well. Just to explain a little bit what you guys are seeing as we dive along and explore, the particles in the water are what we call marine snow, and they are nutrients for things in the water. We're seeing a lot of it, which we're hoping means very productive waters, and hopefully that means we will be finding a lot of organisms as well as we explore the area. I think we're seeing a sea pen in those organisms that they keep their stock in the sand and stabilize themselves as such. Another thing you guys will be seeing is a lot of sargassum at the seafloor. It is a lot of sargassum at the surface right now. You'll also be seeing a halosaur in the top left-hand corner of your screen, which is another deep sea fish. There's been a few of them on this expedition dive so far, but they are always cool to see. Anything is cool to see in the deep sea. But yes, you'll also be seeing sargassum. There are large mats of it coming into the Caribbean waters right now, and they eventually, some of them, once the bladders of the sargassum, which are little bubbles, have been weighed down, they will eventually come to and sink to the bottom of the ocean and provide nutrients as well as homes. Okay.
Little collections, if you see something coming up, we're still looking for sponge, but we know another sponge. We have sea pans, sea starring blood, see if there's a sea pans coming in. Sticking out. Stop it. It's going to be hard to get them because you know they're kind of slimy.
And another sponge.
Yeah, like I say, that is a collection. We'll do a little bit more of that, and then now we're going to transect. So
Okay.
then we'll probably be doing another transect, we'll just running it through the collections. Yeah.
So look out for seepage and
One minute
seeds.
or something.
Okay.
Maybe give it 5 minutes or so, and if we don't see anything, then we'll stop
Okay.
and turn straight south.
Straight out. No problem. Is that one on the top left? Yeah. Yeah. All right, guys, so what we're doing right now is we have found a sea pen. As a result, we're going to actually take a sample here. So what that will look like, if you see right now, you're seeing lasers are on. Lasers are 10 centimeters apart for the 2 dots. And we'll get close to it, get as many images as possible of it in situ, then we'll collect the sample, twirl it around as well so you guys see it a little bit closer, and then we'll put it into our bio boxes. So we're going to start that process as it settles a bit in the water.
While we're waiting for that to settle, we have had some of the sargassum weed going past us all day in some rather large clumps. Right now it seems mostly kind of these little dispersed bands of it, but that's just floating past the ship. See, it's kind of all over out there. There's some larger clumps headed our way again. Oh, is it?
as we wait for the sediment to settle a bit, I can explain to you guys what the sampling looks like. So essentially, we will let all of the sediment settle. We'll get as many images of the organism in situ, meaning in its natural environment, multiple angles, very pretty pictures, and then we'll collect that sample, to a close-up of it, rotate it in front of the camera, and then put it into our bio boxes, which will be on the porch of the ROV. And those samples will then be brought up to the boat later at the end of the dive and will be preserved.
Continue east to try and find another one.
see someone asking about the, they said the stains on the water, right? You can see them out in front of us. That's the seaweed that we're talking about. The sargassum.
All right, everyone listening in and watching right now, sediment is taking a good while to settle, so we're actually just going to do another video transect instead. We're going to turn the ROV, head south for 200 meters, recording the entire way. In those video transects, we do not stop the sampling, we do not stop to zoom in on things. So if we see something cool and we've gone past it, that is why there are transects that must be continuous.
Yep, if we see something
Right,
cool
so
on the
we're gonna
transect, just wave as we go
set
by.
a waypoint now and head south with the ROV, doing that video transect. We will hopefully see the cool things along the way, but we will not be stopping for any samples or to zoom in or out on any of the organisms during that sampling, during that transect.
Got it, Ray?
If you guys look, you're actually seeing a ray or a skate that is on the seafloor right now. We're gonna actually ask the pilots to zoom in on it so we can all take a look. That is absolutely gorgeous, everybody. It is a skate, confirmed. It is a skate from one of the team members. And then when you're about to stop All right, we're gonna have to say bye to our skate friend, and we're gonna begin on Transect, heading south for 200 meters. It's very sedimented. Okay, so first make sure they zoom out. The laser's on and 1 to 2 meters above. Lasers on, or as zoomed out as possible without seeing anything additional on the porch. Yeah. Alright. Starting Transect now. As we're going along, we're seeing a sea cucumber on the floor, sargassum as well on the seafloor. There was a sea pen earlier. We unfortunately didn't get to collect it.
New waypoint. You're gonna be heading south.
Guys, we have a ghost shark. Ghost shark observation. Again, this is a transect dive, so we're just gonna go past it as we do this video. But what an amazing find. We have a halosaur as well at the bottom right of your screen. Those are typical deep sea fish. A sea pen as well.
I'll take a look in case they get distracted. Yeah, we will find check it. They might tend to do it with the
Yeah.
Yeah, just
Yeah. As we're going along this transect, this is another glass sponge, guys. Again, these are video transects. We don't stop or zoom in or do anything. We're just focused on a video for the length of our transect, in this case 200 meters.
This guy's not working right now.
It's working.
Well, I was just trying to make sure it is on there.
There we go. Perfect, thank you. Yeah, if we could hover between the one and the one and two. Yeah. As we cross over here, we're seeing shrimp, sea cucumbers, and starfish again. As we go along for this transect, I guess I can just give you guys some background on this expedition. So this is a deep one. This is a Trinidad and Tobago dive stream that you are on, and this exploration is the first of its kind Not in that our waters have not been explored before, but this is the first time it's being led by primarily Trinidadian scientists. There are 25 scientists on board, 16 of which are from Trinidad. And our waters sits on the continental shelf, so there's an extensive mesophotic zone that is 30 to 199 meters, which is beyond our scuba depths. And then the deep ocean is 200 to 4,000 meters, and that in total makes up 43% of our waters. So as a result, less than 0.001% of our waters have been explored. So this expedition is really going to inform everyone about what there is. There was a paper published earlier this year by a couple team members, Jamie Lee Luchin and Dr. Diva Amon, and they were able to pull from the literature and from previous expeditions that there were about 451 species already observed in our waters. So with 30 days ahead of us of exploration, we're hoping to find and add to that as well.
Plus the 100 meters.
Thank you. On your screens now, you'll be seeing a sea star, some sargassum, and another halosaur. Actually, there is also an urchin next to the sargassum. For anyone now joining in, we are currently doing a video transect, so we're just 1 meter above the sea floor, and we are taking a recording, a continuous recording for 200 meters. We've passed the 100 meters so far of our 200 meter length. You're currently seeing a little shrimp underneath. So as this continues, we will, once the transect is done, we will continue exploring, and potentially if we see any specimens that we would like to collect, we will get them and put them into our bio boxes. On your screen now, you'll be seeing a sea pen to your far left, a sea star, and another fish. Oh, and the shrimp swimming past. Is good. Oh. Oh. Keep going. I think we have to keep going.
No, it's
It's a civil pump.
it's a
Yeah. Wow.
It was a sea cucumber, guys. We just had our specialist on board tell us. And I am just as sad as you to keep going on this transect because we would love to watch it further, but we have to finish our video transect. We have another sponge on our right hand side. That's 200. All right, we're ending our transect now. Yeah.
And right over a sponge.
Yeah, we want that sample.
Surely.
Do you want this?
We ended the transect.
Transect is down here. Do you want the sponge?
If you want it.
Yes, let's collect.
All right, and now we'll be doing a sample collection, guys. We're collecting this sponge. Yeah.
And he did, oh, wait.
No, I think no. He said one Niskin and that was another part two. Oh, just in time. beauty.
Isn't it gorgeous?
Is that always the case? We always do
Do you mean these semantics?
the xanthids, yeah.
Almost always.
There's always on the store.
Always on the side, yes.
If we could just zoom in and yeah.
This is called
Right. The scientific name of this sponge is Hyalonema.
Sponge wing.
Yes, we have a sponge specialist on board who has just informed me. So we're just going to take as many images of it as possible in situ, then we'll collect the sample here and put it in our bio boxes. We want to shoot in this thing?
We have one. There's two actually. We'll try to. Not for this person, but on
And can we find one
before
we
collect.
Before
I think, no. Let's looking through. I think it's maybe this one, but there are no.
So what we're doing now as well is we'll be firing off one of the Niskins to collect a water sample before we do the collection of the actual sample itself.
But they're not pictures of it, but like the things are already
it's
there.
here, yeah, and look,
Yeah,
it's in
so I mean, potentially would be.
yeah, good. It's a little more pointy.
The nose? Yeah, but I mean, it's like, it's a drawing, so you never know.
Yeah, who knows?
Did you get like
For the
close-ups?
Niskin.
Yeah. I don't know if it is.
For the Niskin, then you can right?
Yep. We're moving camera so we can get the Niskin.
It's organizing this thing, yeah. So yeah.
Alright, so we're changing views so we can fire off our Niskin first and then collect the sample.
Start with three.
Or tree. It goes this low, right?
Yeah, should.
Yep. Was Niskin too?
Yes.
Right. Niskin sample is just good. And now we're going to collect the sample itself.
Is distance good for collection?
Did you guys get as many pictures as you need, or you need to zoom in again on that?
Where's it gonna go? Buy a box.
Firebox. Could we put it in firebox?
Which one?
Firebox 3A.
3A?
Yeah.
Okay. I can grab it first.
Oh wait, no. Can we do? We need a. Sorry, you need a total one. Yeah. One of these. So, can we do firebox 1C?
1C?
Yeah, that should be tall enough. So what they're going to do is grab it by the stalk, which is actually very strong. I believe Shirley, who is on board and our sponge person, sponge specialist, has said it is as strong as fiberglass. So.
As long as fiberglass.
Mm-hmm. Do you see in it? And if we could just bring it to the camera and twirl it a bit so we can get close-ups. It's coming close to the camera so you can get as many close-ups.
Oh, yeah. Yeah, perfect.
Oh, wow. Gorgeous. Let me know.
Sponges are beautiful.
Are there enough close-ups you can... Do you want me to turn it to the side or anything? If we could maybe turn it to the side a bit or add the angle of the camera or anything like that, just so we can get multiple views.
Can you do that?
What's the question?
Sideways? Sideways is from view, yeah.
Yeah. All good? All right. We can put them in the box now. Bio box 1.
1C.
We've gotten all our pictures now, so we'll be putting it into bio box 1C. What you're seeing right now is they're extending the porch of the ROV where all our bio boxes are, and we'll be putting the satellites once.
You want them in the same container?
Separate. Okay. Can we do one?
Separate.
Okay, can we do
Not the
bio box 1?
Isn't, no, there's the other side of this.
But that's where we're going.
That's where it's going.
Yeah. Okay.
You're doing 1A, right?
1C still.
1C, 1 is this.
Sorry about that, guys. Thank you.
Medium size now.
Sample of the trick collected. Who's next? Okay. Are we just gonna expand it in?
Yeah, what is it expand?
I think there's a sea pen there.
You can try elusive seepage.
Yeah.
Yes.
Okay, then it's working a little bit. If we have a sea pen,
Well, you gonna have to tell me what is a seepage, but if I see it, it's
It's a little bit of stop and just one and a stop.
all right.
There was one nearby when we planted it. I want to say if we just turn the pan camera on, we might see it. Now that we've collected the sample, we're going to just explore the area and see if we find any other specimens to collect. No, I just wanted to discuss with you.
I'm gonna bring some stuff for them.
And then these are the you can also talk about you can answer questions in the chat there. And then if you have to do the Transect video, you want to check out altitude to 1.1. We try to do in later as part of that discussion.
And how you decided, or you decided to do the Transect?
I can decide. And they'll tell you the length of the Transect. So you want to take the waypoint and start the Transect and the end of the Transect, and maybe every 100 meters.
So do you, the waypoint, just tell us up to the waypoint. So every 100 meters is, and how long is the Transect?
I will drive it only 200 meters, that's what it said, some are 500, so we should discuss what kind of be
When you say the waypoint every 100 meters, do you mean
like just have it put
of
down
something in that 100 or at
100. Just so we can follow the line.
You see how on the left side?
No, just have fun with it.
Oh, right.
For
I think it's kind of a fisheye.
So I think also the go shock of the game. We have 5 minutes. Alright guys, we're exploring now, so if we see cool things, we will be getting close and taking samples if we can. There goes a little shrimp.
Oh, really? That's the stickers.
Stuff. Is that it? What? Yeah, there's a seat one on the to the left. That one? We want the left one? Yeah. Okay, we're gonna take that one. Yeah. Alright, we found ourselves a sea pen, which we've seen a few of them already, but we're gonna actually take a sample of this one.
That's an exciting one. Is this the ones that disappear?
Yes.
Yeah.
Alright.
Hopefully we get it this round.
Sneak up on it, Rafi.
The distance is a bit.
Which box? Alright, so we're gonna get as many pictures of them in situ as possible, and then we'll, when we get in the call, we'll take a couple more again. So we zoom in, maybe not get firebox. quiver? Okay. Where are the quivers?
Just on the quivers.
Okay.
But the numbers are flipped. So for some reason, it's dipped. This is 1, this is 2, 3, 4.
Okay. So when I tell them the correct version, not the wrong. So they're seeing
They're saying
2,
yes.
1. Okay. Oh wow! Look how gorgeous that is. All right.
Gorge.
Stunning. Do you think we could end this? Do you want me to see if we could change the article? Want to change the article? No, the amber of the pictures. Okay, so we're gonna put it in a quiver. So if you could put it in quiver, this is wrong, but this will be quiver 1.
Collection?
Yeah.
Yep.
Number 2, you said? Yeah.
It will be
Yeah,
1.
but it's gonna be choose one.
Is it real out there, you know?
Extended already? Oh, yeah, you need to open
And when it's in the arm, if we can rotate it so for pictures as well.
it. All the way?
No, it's good. So you want this one with the orange, right? Closest one to us.
Yes.
Right, just watch my arm on this screen and make sure I don't hit this reality. All right, I need to focus on that. Two.
As they're doing this collection, I'm actually going to switch over my shift with my colleague Robert. So Robert will be joining you guys for the rest of the next hour. Bye, everyone.
Hi everyone, I'm Robert Cassidy, PhD student at the National Museum of Natural Sciences in Madrid, taking over for Charisse. Thank you, Charisse. Okay.
Each one is the opposite.
1, 2, 3, 4. Okay. Cool. And you guys just make sure to take some photos. Or to roll spin.
photos? Can we store it? What's the...
Yeah.
Did you guys get that?
Pretty good. So that's going quicker too.
Yes, it's
Yeah.
gonna be... It's named a tube, but it's
okay,
one.
sorry, one. Yeah. Thank you. Okay, so that is our 6th biological sample and 9th sample on this dive so far, if you include water from the Niskins. That was a sea pen.
Just flying around.
I think so. Yeah. Starting to translate now, or just flying around? Yep. Seeing what we see. Okay, so now we're back up. We've got, looks like some more octocorals on the right. And we're just gonna fly around for a bit and see what we see. As some people have mentioned before, one of the goals of this trip is to collect environmental DNA samples. And that just refers to samples of the DNA that is loose in the environment and has been shed by basically everything living down here. And so the point of that kind of sample is to collect data that you can't typically get through visual surveys or through typical taxonomic surveys. Oh yeah, look at that. What is that? That's a nice, looks like octocoral, I think. Oh, okay. Looks like we might have some barnacles actually on this. And it looks like a Venus trap anemone on the top as well. Can we collect this piece?
Yes, we can.
Yeah. And if we could take, just have some time to take a photo first, I'll tell them to get some grabs.
You guys getting photos?
If they want to plug anyway, then just set down.
Okay. Yeah. All right, so we're going to try to grab this one as well. Yeah. Okay. And this is eventually going to go in Coral River 2. Yeah, so you can see those barnacles. It looks like they're feeding with these little arms that reach out and grab detritus from the water. And you can see on the kind of middle bottom left, there's an anemone as well on the stalk, just closed up.
Happy for us to collect?
Yep, I think we're good, right? Yeah, we're good, guys. I think not the seaweed. Just, yeah, that's good, I think.
Picture this up, correct?
Yeah. That's what I'm picture this too. Yeah. It's just like, I don't know, it's just more and more stuff the more you look at it, right? It's crazy. So on this one sample, we've got multiple anemones, a bunch of barnacles, and it looks like octocoral as well. So that's a pretty exciting find for us. That's great. Yeah. So that sample is just a great example of the way that structural invertebrates like corals and sponges can provide a habitat for lots of different types of animals in these deep sea areas especially. Okay, now we're back up, flying around. Just gonna see what else we see here.
Oh no.
Are we heading to the south now?
Yeah.
Yes. Yeah. So each of these samples we collect are actually going to be used for a variety of different purposes. A lot of them will help form, they're all going to help form the baseline of biodiversity information that we have in these waters around Trinidad and Tobago, which is great because this is a first of its kind expedition in this area. Each of these samples is going to have, we're going to have museum vouchers for further taxonomic work in the future. We're going to have genetic data on each individual sample, which will give us, information on, maybe where it comes from, what it's connected to population-wise, and help form a database of biodiversity in this region based on genetic data. Ooh, that looks like another sponge, I think, along with something else on it.
Did you want more sponges?
Yeah. So it looks like one of these glass stock sponges, as well as I think 2 anemones attached to it as well. Yeah, I think we can keep going.
Do you want the sponge or not?
I think it's okay. Yeah.
You first one to see this guy?
So one of the main reasons we're actually so interested in sponges on this trip is that they actually serve as natural filter feeders, and they can pump so much water through their tissue that they actually capture the DNA from other organisms in the environment around them. By sampling that tissue, it's possible to sequence DNA from not just the sponge, but from the entire community around it. And so that's what we're going to be doing on this trip, not only in the lab when we get back, but also on board a little bit. And that will help form the, oh wow, it looks like a huge shrimp in the top right. Yeah, we could try. Should we try to slurp this one? Possible?
The second one is
Second one. Do you know what kind it is? So we're gonna try to collect this one too. See if we have any luck.
Yeah, we'll give it a go. You want some good video first, right?
Yeah, thank you.
So you can sit, you know, if you want.
no, we can. But we see the second one is.
That is massive. That is huge for a shrimp. Yeah.
Yeah, I wonder if it's definitely.
So those two laser points are 10 centimeters apart for scale. So this is a very large shrimp.
Okay. Want to go for the start? Or?
Yeah, that'd be great. I think we're ready.
So let's do 0, 1, I think is going to large. No. Yeah, 1.
Let's move forward.
Oh, it's on 1. It's on 1 right now.
Chamber 2. Okay. So we're doing chamber 2 for this, please.
You can. I would recommend 1 if possible, though, because that's the biggest mesh. That'll give us the most flow, because this guy's probably going to try and run away as soon as we can. Okay, we'll put it to it. I mean, you might get away anyway. You know how these guys are. The minute you spook them, they... Okay. So you want 1 then?
Or 2. Okay. So 1. So we're trying to use the suction arm to capture this guy because they're pretty fast and they could just easily escape the claw.
Sometimes you don't want to do it before because they can feel the suction and freak out. I'm looking behind this guy because they always tend to go back. But I think he's going to go away. I think it's too big. I'll just try it.
Don't have it.
Pretty long device. Keep trying the flows.
Light.
Once you get into full section, my bungee is going to put his antenna and try and get down. So that's where the suffering jumps up.
Right, can I start then?
Go ahead. Fly. Suction is right now, right?
Yes, it is.
Oh, and he got away. Good try, guys.
Guys, that was never gonna work.
Diva's saying it was never gonna work. It was brave.
Search for a bigger one now.
What's that?
Let's find another bigger one.
Yeah, let's go for an even bigger one. Okay, yeah. So you can just keep flying around, I guess. See what else comes up. Looks like there's something over there, it looks like. Oh, okay. Oh.
Well, I mean, subsample.
Yeah.
And the thing is, when we're going to transplant, we can then once it changes.
Looks like we have more barnacles here. And looks like we have another glass sponge here as well. Or can you zoom in more on that?
Wow.
Wow.
That's cool.
Yeah.
I think we should get this one. It's different from others. I think we want to get this one, guys. Yeah. We could zoom in for some pictures. This looks different, right, Troy? Yeah. I was thinking we should collect this one. What do you think? Yeah. Okay. So if we get some pictures here for a second, zoomed in. This is another stalked sponge, a little bit different from what we've seen before. And it looks like it has zoanthids on the outside there. You can really see with these why collections of sponges are called sponge gardens, because they're just very beautiful, you know, like flowers.
What is it, Shirley? Sticking behind that. What is it? Is that a little arm of somebody? Somebody's in there. Somebody's
You know, it could be a crab with that on its back.
Wow.
Except for it has a whole thing.
It looks like some kind of crustacean's claws poking out the side there, but not entirely sure why it would be. If it's from a live crab or shrimp or from something else. Yeah, I'm going to. Could we actually do a Niskin before we collect, guys?
Yes, we can.
We could do 4. Yeah.
Use the camera.
Okay.
Yeah.
That's what those are, you know, on the start itself. Or on the start.
We're getting a Niskin here too. We're getting a Niskin here as well in case that is
All right, where are we putting this?
This is gonna go, I think, in 3A.
Try.
Too big. Yeah. Yeah, 3A. We try 3A.
Go.
I think it should fit.
Okay, let's see what's fine there. I just noticed it was going to run on the back of that sponge. Oh, what was that?
3A. Yeah.
It's in the front one here.
Yep.
Oh, somebody's back there. Yeah.
Oh, wow.
Look at that. Crazy. Wow.
Wow. Look at the roots on that. Those are spicules.
Is that a...
Spicules,
It's
those
a...
are silica spicules. Yeah.
So,
They're not
Wow.
from that sponge.
no. From a different sponge.
I think the spicules are from a different sponge.
So these roots we're looking at are not roots like a plant. These are actually spicules from the sea sponge. And spicules are those little bones that form the body structure, that give it kind of a harder structure. But these ones are adapted to root the sponge to the ground and are...
It's not enough.
Let's go in the back. Okay. Can we actually change to BB1A? It looks too big for the front box.
Yeah,
1A.
Sure.
The big one.
But then you may have to share it with something else.
Do you all want any more video of this, or you get to put it in?
Yeah.
I think we're good to put it in.
I wanna know.
Wow.
The lower line.
There was a sponge right on this.
So it was one of the glass sponges that made the stop, you think? And that,
Glass sponge, and there's, I don't know if that's a glass sponge or if it's a demo sponge. It looks like it might just be a glass sponge. Those spicules are very different.
So our resident sponge legend and expert, Shirley Pomponi, is telling me that her theory is that, so this is a very unusual sponge, right? It has some of the markings of these white glass sponges we were seeing before, but it also has a different color, and it looks like these spicule roots may have come from a different type of sponge entirely, is what Shirley is saying. So that'll be very interesting to see back on board.
Yeah. Yeah, there's a spot on there.
Like, is that the... That one came off of it.
That one came off it,
Yeah.
but a good one came off it.
The lowers.
I can see a squat lobster swimming away there.
Yeah. Do you think you get that perception? Squat lobster. Do you think you can try deception then? Yeah. Okay. It's a little bit smaller than the other one, right?
Oh, it's a... It looks like an anemone.
Look at those tracks.
If we could zoom in and get some photos first, that'd be great too.
Very surprised.
It's looks like it's covered with little tiny zoanthids maybe too.
Just fell off the roof there.
I think he's freaking out right now.
Fell off the roof. It's very bright out there.
Yeah, pretty good.
Which photo?
We can try what we did last time. I guess it'd be
The first one?
first one. Yeah. You want to do one or two for the section? We can try to get the squat. But you think one or two for the buff? We tried two?
Two?
Yeah.
Sorry.
So we're going to try to suction this one as well. See if it doesn't get away from us.
Suctioning now?
Yes, it is.
And we got him.
Abducted.
Nice job, guys.
There he is. He's in the jar, so you can kill suction with duct tape. Yeah.
Very nice.
Any other one in there? Serums?
Okay.
It's neat, right? Yeah, really neat. Okay, and that should be underway.
Knock off the lowers a bit.
I'm just.
I cut everything, all the old books in there, but I still haven't.
Oh, you probably got trimmed, so press that button right under that.
Thank you.
Okay.
Because there's a lot of buses.
Yeah, so what I was thinking is, once we come home, then we start doing some classes.
Yeah. So yeah, at least 300 meters, 1 meter, but
I think it should be high, because we're not sure how
trying to find
well,
habitat.
yeah, exactly, so I don't think
So we saw before.
so we take on, we can do like a 500 meter non-stop. So we have 10 minutes, let's say the same thing.
That is
Keep starting in the next 10 minutes, let's say same thing.
with our Venus
Fantastic,
transect.
I don't think, but then. Okay. Is he worried about too many for processing?
For the first day.
Could you zoom in on that white-ish
Those,
one? Yeah.
Yeah, and we have a few samples, but there's definitely associates.
Oh, for
with
sure,
samples.
for sure. So we'll spend maybe 10 minutes sort of looking around, and if we find something spectacularly different, then we can sample it, and then at 3:30 we start doing transects again.
Okay. Sounds good.
It's another sponge.
Yeah, it's right.
We have 5 sponges.
Yeah, we don't need to collect it. We don't need to collect on this one, guys. So.
sitting right on a brittlestone.
It is, yeah.
Landed on it.
It looks like it just got squashed
Yeah.
by it, doesn't it?
Right? Happy?
Yeah, that's good. Thanks. Yep.
I think it's a glass sponge again. So when we say glass sponges, Exactinalia is the one on the right. And you can maybe make a note that I did not have a stock on my the other ones. That's all I'm going to say. Sure.
I need like it's a no name.
It's true.
So it looks like we have another sea pen here also on the left, and a sea star as well. We don't need to zoom in all of it. That's okay.
Those are easy, right?
Yep. Awesome.
Oh, I see.
So we have a waypoint drawing towards, or
No,
just
we're
explorers?
See our parallel parallels the original line. So waypoint is down south. So once we get over, we put the flag on just due south.
Going the straight line in the same
Straight
direction?
south, yeah. And it could be here. But then we transfer it to the yeah. Oh, 30 meters.
Your leading waypoint.
30 meters.
Yeah, yeah.
Okay, waypoint. Is that the one that's just outside the red circle?
That's the one.
Yeah, so you know, you don't have to move.
Looks like another shrimp on the center screen here.
This we definitely don't care about.
Should we try again, or
No.
we're not gonna try to get this one again? They're too nimble.
These are not even working.
Another fish on the right here. Not sure what kind of fish.
Try that.
I think we have another C pin down here on the bottom. Big sea star. Oh wow. So we got a ctenophore, I think, floating by. You can see the light shimmer across its cilia. Very cool. Okay. Ready?
We are ready. No rush.
Sure. So I think Anne said they want to do another transect starting now.
Another one?
They want to do another transect starting now.
Yeah. Yeah.
Yes. Cool.
Say bye.
Okay, thank you all. Gonna pass you all over to Beatriz Naranjo.
Good.
You? How are you doing?
Really good. Yeah, but I would like to do that concept like 500 meters long. You can put waypoints to divide that in 100-meter segments. That would be amazing.
No problem.
Make my life easier analyzing it.
Alright.
So it will be like, yeah, 5 chunks of 100 meters.
Straight south.
Straight south, yeah, please.
And from here I need to go align with the waypoint.
I mean, I think I know because it's really like sedimented. I mean, like clouds inside. Hello everyone, my name is Beatriz Naranjo. I'm a biologist from Costa Rica. I'm an independent consultant collaborating with the University of Costa Rica and other organizations. And I'm really glad to be here supporting the a local team from Trinidad and Tobago who has done an amazing job preparing for this expedition. It's a big moment for everyone here. And definitely this expedition will increase knowledge, build capacity, and support conservation of these amazing environments that we found in the deep sea. Right now we are going to start some video transects where we try to keep the distance from the ground or altitude, the speed, and we also use lasers that you will see in a minute. So this transect is going to be 500 meters long in total. So with this transect we aim to being able to better quantify all the biodiversity that we will be seeing.
Yeah. 200, right?
Sorry?
It's 200 total, right?
500 in total.
What's 500 in total? Okay.
Yeah. It's like,
Yeah.
yeah, we already have like 20 samples for today maybe, so we will take a bit of time for the images.
All right. And you want to mark every 100?
Yeah, I mean, if it's easy for you to mark all of them now, we can, or we can just start at each 100 just to flash lights.
Okay.
But 500 meters in total. Buenas tardes, mi nombre es Beatriz Naranjo. Soy una bióloga de Costa Rica. Colaboro con la Universidad de Costa Rica y diversas organizaciones que trabajan principalmente en el Pacífico tropical oriental, estudiando nuestros ecosistemas de mar profundo, y hoy tengo la fortuna de estar apoyando y acompañando en esta gran aventura en Trinidad y Tobago al equipo local que ha hecho un gran trabajo. Es la primera vez que una expedición se realiza en estas aguas de Trinidad y Tobago y que sea organizada y liderada por personas de Trinidad y Tobago bajo el liderazgo de la doctora Diva Amon. Han hecho un increíble trabajo de comunicación, tanto antes como durante la expedición, y estoy segura que todo este conocimiento que se va a generar durante esta expedición va a incrementar muchísimo todo el conocimiento que se tiene de estos ambientes. Así que muy feliz de estar acá y apoyar a este increíble equipo que está compuesto por principalmente científicos, científicas de Trinidad y Tobago, otros países del Caribe y también de otros países como Canadá, Estados Unidos y Costa Rica, por supuesto.
Good to go?
Yes.
Good to go?
Good to go. So start transect number 3. Yeah. Yeah, so you go to the transect, and you do transect 3. So you just check 3. So start. So we are right now exploring the western part of Trinidad at almost 1,000 meters depth. And yeah, I just want to call out all the big effort that, thanks to Schmidt Ocean Institute, all of our region has got with all of these expeditions happening through the south. we started exploring the southern part of the continent since 2023. And this vessel, the Falkor 2, has been around different countries around the Pacific side, and then went to Argentina, where they have amazing expeditions, coming up to Brazil, and now Trinidad and Tobago.
Bridge, RV.
RV bridge, go ahead.
We're starting a 500-meter transect.
Okay, starting a 500-meter transect.
So during these transects, we try to keep a speed of between 0.1 0.2 maximum meters per second, and trying not to stay above the ground beyond 2 meters, so we can keep better results using these lasers that you see on the screen that are 100 meters apart. Take like 1 hour. Yeah. So a while ago we were seeing a lot of sea urchins, maybe 1 hour ago and before, and we have also been finding sargassum algae, which is a macroalgae, and we have been seeing blooms of this sargassum on the surface, and some pieces of this one that we have just passed. And all these sea urchins might be here really abundant because of that reason. They are known to feed on different kinds of detritus, and also this kind of algae that is now in the bottom, falling from the surface. So with these transects, we want to understand a bit all those changes that might be happening. You might know that all these sargassum blooms have been really common, probably since 2010, 2011, and they have been increasing for different reasons. Contamination is one of the reasons, a lot of nutrients in the water, also dust coming from the Sahara that brings a lot of metals like or compounds like iron and phosphorus, and this help this algae to grow really fast and produce these blooms. And of course, the warming of the ocean is other reason. So they have a lot of impact, negative impacts here in the region, especially because many of these countries rely on tourism, And as you might have seen some images, they get to the beaches, so it's been a problem for fisheries as well, and coral reefs and so on. So as you can see here on the left of the lasers, which is a part of the of this algae, a sargassum, all the impacts are coming even to the deep sea. So that blue one is the first waypoint, the first 100 meters, right?
It's going to be around here.
Ah, okay. Oh, it's there. I see. Okay, okay. So yeah, you can just like do some lights
Okay.
to remember. So there's a lot of institutions involved with this expedition. We have actually here as loggers students from the University of West Indies, San Agustin from Trinidad. Also people from the Institute of Marine Affairs of Trinidad as well. And we have two researchers from the Harbor Branch Oceanographic Institute from Florida, who are studying sponges and a really new method to preserve them for the future, preserve their cells. So their research is really important on sponges because, as you might know, there are different animals even in the deep sea that have really interesting chemical compounds that might be helping the medical research. So you can find different medicines in the deep sea. So that's why one of the reasons why they are here, collecting sponges and making a collection of their cells that can remain for future generations. We also have people from the University of Calgary who are conducting heat flow measurements and studying how the crust is losing heat, which is really interesting because you can find even hotspots of biodiversity when in places where this heat is more frequent or more intense. For example, octopus nurseries, skate nurseries. We also have one researcher from the Halls University in Canada and from the National Museum of Natural Sciences in Spain. So it's a really diverse and motivated team. We are putting a lot of efforts on getting new people excited about the deep sea. A lot of students who are pursuing their their university studies. So it's going to be an amazing opportunity for the region to increase all this capacity. This is one of the glass sponges that we already collected. This is a glass sponge, and you can see the stalk all covered by zoanthids. And I'm sure if we did a zoom, you will get to see a lot of tiny animals also living in there. So sponges, as you can see, this is a really flat environment. So when you find corals or sponges, they are one of the main architects providing three-dimensionality to this environment so other animals can live in. Oh, shrimp. This might be the most common fish that we have seen already. It's a halosaur, and it's being like really abundant. Ya que veo algunos saludos desde Argentina en el chat, quisiera sí enfatizar que desde que el Falkor ha estado explorando la región de Latinoamérica y el Caribe, el conocimiento que tenemos de mar profundo ha incrementado considerablemente a un ritmo muy acelerado. Todas estas expediciones usando el ROV Sebastian, y que ha visitado diferentes países desde Costa Rica, Ecuador, Chile, Argentina, Brasil y ahora Trinidad y Tobago, tiene un impacto sumamente importante en cómo podemos manejar y conservar este patrimonio que es el mar profundo, que compone la mayoría de nuestro planeta y la mayoría de nuestras zonas económicas exclusivas. En el caso de Trinidad y Tobago, estamos hablando de un 93% de su zona económica exclusiva se encuentra en el mar profundo. Y esta es la primera expedición que lidera un equipo trinitense y donde están participando diferentes instituciones del mundo, pero 16 personas de todo el equipo científico es local, lo cual es todo un logro, y que tenemos entre científicos experimentados a estudiantes, personas que están terminando sus maestrías, buscando doctorados y que se están enfocando en el mar profundo. Species, que es la ONG que ha colaborado con la organización de esta expedición, ha hecho un increíble trabajo comunicando sobre el mar profundo, tanto antes como por supuesto durante, y como van a ver todas estas imágenes que estamos recolectando, todos estos videos y todo el conocimiento conforme se vaya procesando científicamente, va a apoyar muchísimo esos esfuerzos de comunicación, de educación, de sensibilización sobre el mar profundo y por supuesto informar todo lo que está pasando en Trinidad y Tobago, y no solo para Trinidad y Tobago, sino para toda la región caribeña. Como ustedes tal vez sepan, estudiar el mar profundo es sumamente caro. La mayoría de nuestros países no tiene acceso a estos equipos. Y como hemos visto, por ejemplo, en Argentina, que las expediciones fueron virales, hay muchísimo potencial para que no estropeemos estos ecosistemas que estamos viendo. Esta es nuestra primera inmersión de casi un mes de expedición que vamos a tener. Así que estoy segura que vamos a tener descubrimientos sumamente interesantes. Mencionaba hace un poco que tenemos, por ejemplo, a bordo investigadoras de Harbor Branch de Florida, de Harbor Branch Oceanographic Institute, que se especializan en el estudio de esponjas, que es un grupo sumamente interesante porque contienen compuestos químicos que pueden ser las medicinas del futuro y que ya están demostrando serlo. Ahorita a la derecha de los láseres tenemos un trozo de sargazo y un erizo a la izquierda. Aquí tenemos otro de esos erizos. Hemos estado viendo muchos de estos erizos que se alimentan de detrito y que también se alimentan de estas algas, que como sabrán han habido blooms de algas a lo largo de la región y que están llegando hasta acá al mar profundo. Ahorita estamos haciendo un transecto de 500 metros, lo que nos va a tomar bastante tiempo. En estos transectos intentamos mantener una distancia del fondo constante, así como una velocidad constante. Estos láseres que ven son de 10 centímetros y nos ayudan a medir organismos y a calcular el área que estamos viendo. Con estos transectos podríamos volver en algún momento, comparar cómo han cambiado esos sitios y los hacemos exactamente en los mismos sitios, y también nos permiten comparar entre lugares. Estamos pasando una pluma de mar, un pepino y un pez alosaurio, que han sido los más abundantes que hemos visto.
Problems, pressure problems, you know, here. You can see the pressure, right? That's pretty good, right? But sometimes it will drop like 1800, 1600. If you look at your thrust and you see that pattern, it arms going to the side. That's what it was. So you got to move on. That way, and then it should be like that. Well, what quarters on this?
Were you here when we saw the ghost shark? Were you here when you saw the ghost shark, the chimera?
No.
You're not here? Okay. We saw two already.
No, I was flying for one of them. We've seen two, right?
Yeah, two. How often do you see them, you think? Not all the time, right? So here we are seeing a Venus flytrap anemone. I think that's the common name in English. Una anémona atrapamoscas, Venus atrapamoscas. More halosaurs. And more sargassum. So less than 0.001% of Trinidad and Tobago's deep waters have been explored. So with these expedition, we aim to cover new places, as this one where we are right now. We are not right now at 1,000 meters, and some a previous cruise back like 10 years ago found like at least 85 species just in 3 days, potentially 5 new species. So if we are hoping to get a lot of new information, species, new habitats that have never been seen before. And there's a recently published study based on literature and records in different databases that documented 451 species that have been officially reported in Trinidad and Tobago's deep waters. But it's estimated that more than 1,600 might be present. So all that remain to be discovered. Another glass sponge. So for those asking about the lasers, they are separated 10 centimeters between the dots. Here we got another anemone and a sea star. On top, what it seems to be a rock, maybe. We are finding some crinoids now, which are echinoderms. Earlier, we also saw 2 chimeras, which was really exciting for us who love sharks, ray, and chimeras. And they are not common to find, not weird, but it's definitely not a common animal. So every time we get one, we get excited. This is a sea cucumber on the right side. We collect that species. Was our first sample from the expedition, actually. So we are heading south and slowly climbing a slope, which is not really steep, but we are right now at 998 meters. So 8.4 is the end of the transect, the overall transect? Okay. Another sponge full of zoanthids, another sea star, more sargassum, sea pen. We also have in the room, just enter Dr. Shirley Pomponi. She's an expert, sponge expert, and one of the persons here on board who are using these sponges to preserve them for the future with some really innovative ways to preserve them, to preserve their cells. So we are really excited about that part. And so she is looking at all these sponges. We already have some samples to work later. Another sea star. So during this course we'll be exploring below 2,000 meters, and no one has ever explored below 2,000 meters here in Trinidad and Tobago. And we will also be exploring the mesophotic zone, which is between some 50 meters to 200 meters deep. So the depth range for this expedition is pretty wide. So before this dive, the first thing that we made when we arrived on the site was to conduct some seafloor mapping. Right now, the maps for the deep sea in the area are really, well, not high resolution, so we are doing also that part that also allow us to choose the best locations to dive. So there's a lot of mapping. There are other research taking part during this cruise, not just with the ROV, but also we are conducting plankton tows, including ichthyoplankton tows that are aimed to collect fish larvae, and those samplings will allow us to study the connectivity between places and also vertically in the water column, other connectivity with the deep sea, of course. There's also research happening studying microplastics. The vessel has a system to collect water specifically for that purpose. So we have a person involved in that research as well. So yeah, a lot of research coming in the next years thanks to this expedition. What is that? I think that was a 1004 again, maybe. Another glass sponge. We already collected a couple of these sponges before. You can see a leaf on the seafloor. And an important thing to mention is that all these samples that we are collecting will remain in the country. The voucher specimens, that they are the ones used to identify the species, to describe them in case they are new. And that will increase a lot of the local capacity to conduct taxonomic research, which is like the base of all the knowledge that we can get from the deep sea. This is really important because in the past it was really common that people just take their samples to their countries and they did not get vouchers on the home country, and that's where they should be, for sure. We keep seeing sargassum everywhere. And we keep also seeing sea urchins of the genus Formosoma. Sea pen, which is a kind of coral. There's a Formosoma sea urchin on the left, the one I was telling you about. think we keep going south. Relax. That's the plan. I think we keep going south to the main waypoint, but it's below the original from that plan. Yeah, so, yeah. For now, it's, yeah. Argonia Sterid sea star on the left. We already got one of those earlier. Seguimos viendo más sargazo, más erizos que probablemente se aprovechan de la presencia de ese sargazo, ya que son organismos que se alimentan de detrito, de materia orgánica en descomposición. Y como ven, este sargazo está por todas partes. Uno de los avistamientos más interesantes que tuvimos hoy fue 2 quimeras del género Hydrolagus y algunas esponjas que recolectamos. Otro alosauro que parecen dominar completamente este lugar. Alosauro significa, bueno, alo significa mar o sal, y sauro de lagartija, o como los dinosaurios, reptil. Así que sería como una lagartija de sal, lagartija marina, muy abundantes en este lugar. Diferentes especies de camarones. Plumas de mar, esponjas, pequeñas estrellas. Cities close to the wasteland, that will be the end of the transit. And the other, I think we already know transit for now. Okay. So you can, I'm gonna, once we change, I can go and check. But I think you just go south towards the original code point, which is like just the line over there. Okay. You can see the pink one. We run this transit.
We just proceed up to waypoint 3.
No, we are coming from waypoint 3, so we go towards waypoint 2. That's the end? Okay. Shannon, can you log the end of Transect? It will be the same number, but the status end. So we are ending this transect here. This was a 500 meters transect. And for now we will keep exploring, but now we will be able to zoom in anything we see. The end, right? Almost.
There.
Okay.
Almost there. There.
Okay.
Gonna continue heading
Yeah.
south, right?
Yeah, please. But we can stop and do zooms.
Yeah. Pretty charming. There you go. Yeah, we're gonna be continuing
going south, so I put a new waypoint.
Can you see what that is? Or So I'm about to finish my shift here, but it's an honor for me to leave you with Shirley Pomponi, which is the sponge expert that I was telling you about. And I'm sure she can explain way more better than I all the research that they are doing, that it will be really important for medical research. Estoy terminando aquí mi turno. Un saludo a toda la gente de Argentina que nos ha estado saludando, toda la gente de Latinoamérica, de Costa Rica. Es un honor para mí estar aquí y terminar mi turno dándole el paso a la doctora Shirley Pomponi, que es la persona que está a bordo estudiando esponjas de mar y los compuestos de importancia médica que tienen estas esponjas. Así que básicamente el futuro de la medicina. Shirley, are you ready?
I'm ready.
Okay.
Hello, everyone. My name is Shirley Pomponi. I'm a sponge biologist and biotechnologist. I spent most of my career at Harbor Branch Oceanographic Institute in Florida. I'm participating in this expedition as an independent scientist, and I'm really excited to be involved in it. This, in case you're just joining us, this expedition is one to explore the deep waters around Trinidad and Tobago, areas that we've never explored before. We have a team of 25 scientists and one artist. 15 of the scientists are actually from Trinidad and Tobago, including our chief scientist, Dr. Diva Oman. We have 3 other researchers who are based in Trinidad and Tobago, not nationals. One of these is from Barbados, and so we have 16 Caribbean nationals on board. So far, we've been, it's been pretty, I know you probably think this is really boring because we've been going over mud, sediment bottom for most of the dive, and I'll talk a little bit about that in a few minutes. But we have several objectives on this cruise, mapping the seafloor, getting some data on heat flow, mostly identifying some of the benthic, so bottom-dwelling and pelagic or midwater marine ecosystems and some of the biodiversity hotspots. So it's pretty exciting. I'm going to be narrating for the next hour. I have lots of family and friends who are joining. And I wanted to do a special hello to two very special people, my grandchildren, Jackson and Kylie, who are joining from Florida. So I hope you enjoy this. Okay, so we're just finishing what's we call a transect, so just going slowly across the bottom and videotaping, photographing, really, the whole seafloor bottom here, making observations. We have a really amazing database that Bia, the narrator just before me, has put together. So we're making really detailed observations about everything that we're seeing. And now what we'll be doing is just continuing across not continuing anywhere safely. As soon as we've reached the end of the waypoint, we're going to turn around and head south, I think, and then continue across the bottom and hopefully see some more interesting things. So I mentioned I'm a sponge biologist and biotechnologist, so as you can imagine, if you've been following this for the last several hours, I've been pretty happy. We've seen and collected a few sponges, and can't wait to see them when they come up on the surface. So the reason that I'm on board is because we're going to be testing out a new technology, actually freezing, or call it cryopreserving, pieces of sponges, small pieces of sponges, and anything else that we collect, and it's called that cryopreserving, and we put them in a freezer, and they remain alive. So when we thaw them out, the tissues will still be live. And so it's a really tremendous resource for conservation of biodiversity. We're using a very special cryoprotectant that we've not used in the past, and it allows us to cryopreserve using non-toxic solution, and instead of having to put the samples in a negative 80-degree freezer, we can put them just in a regular freezer like you have in your own home. And we can keep them in that freezer until the cruise is over, and when we get back to the lab, we can put them in a freezer that's a lot colder. And did you want to say something?
No, I was going to can you ask the pilots that if we want to be on deck at 8:30 p.m. on deck,
okay,
what time we need to leave bottom?
Okay.
Just ask them. Because it's supposed to come.
Okay. Okay, stand by. I need to ask the pilot a question.
I didn't want to go above Shirley.
So, we will be able to.
On deck at 8:30.
Yeah. Okay. Hi, I'm back. So this might look really, really boring to you. And so if you're used to scuba diving and seeing all these like amazing colors and different animals, when you're scuba diving, this is way different. But don't be fooled by the fact that it's just this, what looks like a really boring seafloor. And we're coming across right now a stalk of, I'm trying, is that still alive? It might still be alive. Stalk of a glass sponge, or it's called a hexactinellate sponge. Yep, it's still alive. We collected a couple of these already, so we don't need to collect this one. But this is really cool. It's the most common sponge that we've seen today. In fact, and we've collected 2 or 3. On the stalk of that sponge, the stalk is made out of silica, kind of like fiberglass, very long. They're called spicules, skeletal elements. And on those spicules now, another animal settled and grew, and it's called a zoanthid, which is like an anemone. So it's living, just in association with this sponge. The sponge it's called a glass sponge. They're commonly found in deep water environments. There are a few places where you can find them in shallow water, like in fjords off of Norway, or even off the coast of Canada. You can find them in shallow water, but generally they're found deeper than 200 meters. And if you're looking carefully, you can see that there are kind of mounds or even tracks along the surface of the sand or sediment. And those are made by animals that are crawling along the bottom or living buried inside the sediment. Every now and then we're seeing some sargassum, so a seaweed, and I think the previous narrators have talked about that. Can we go up and take a look at what's up in that kind of the upper right, please? I don't think we've collected any of this. Let's take a closer look at this. This might be another glass sponge, but I want to just take a closer look at this one. This is really, really nice. I don't think we've collected any of this before. This looks like, boy, it looks unusual because we see all of those. I don't know if those are tentacles from. Yes, I would like. Yes, I would like to collect it, please. Thanks. Yeah, we're gonna collect this. I think it might be a heterotella sponge, a glass sponge. I'm kind of intrigued because you don't see all those, I don't know if you can see all those projections along the lower half of this. And that's actually quite unusual. I've just not seen it like that before. I'm not sure if it's normal or unusual, but I've not seen it like that before. If this is what I'm thinking it is, there's a kind of a sieve plate over the top of it. Oh, this is really beautiful video. And we'll probably, I think we're going to want a Niskin sample. Is it too late to get that?
You want a Niskin?
Yeah, can we
On
get a
seabed,
Niskin?
or you want it up higher?
Higher.
So you want us to get this first, or?
No, get the Niskin first if you can. So we're going to collect a water sample nearby because one of our previous narrators, oops, looks like there might be a ctenophore comb jelly that's floating by. One of our previous narrators is working on what's called eDNA, environmental DNA. So he's trying to figure out if these sponges are filtering out DNA from other organisms that are in the same environment, say fishes that are swimming by or other things. And so he'll be taking samples of this sponge when we bring it up to the surface and extract and pull out and analyze the DNA to see if it's got DNA not only from the sponge itself but from other things as well, because sponges are really good filter feeders. They filter a lot of stuff out of the water. But kind of as a control, he wants to see what's actually in the water that's nearby. So we're going to take a water sample first. It's put into a special container called a Niskin.
Yeah, that's 5. All right.
Okay?
5. Number 5. Please.
Number 5. Okay. It's in 5. Thank you.
Got really good images of the sponge. Really good before collecting.
Yes.
They say no.
So we don't have. We'll take some more. We'll get some more video. We'll get some more video of this before we collect it. If possible if we could get a video kind of looking at the top of it, because I think it has like a little sieve plate over the top of it. So someone on the chat actually said that he spots shrimps inside, and that's quite possible, because if this is one of those sponges, that's a heterotella or euplectella, they have a pair of shrimp who live inside of the sponge. We're going to get some images. We're going to take a look and see. I think it's straight ahead of us. So it'll be good to get some images of those, what looks like shrimp, yeah, on top as well. So this sponge is really interesting. It has on the top of it like a sieve, and its common name is Venus flower basket. And getting down. So of course the sieve has holes in it, and what happens is a pair of shrimp kind of swim in and out, kind of settle in there, and they're living inside this sponge. And eventually, they get too big to leave, to pass through that sieve. So they're kind of trapped in there for the rest of their lives. But because the sponge is very porous and it's got it's filtering water all the time, there's water flowing through it all the time, they're still able to survive. And while we're getting in position to get some nice close-up photographs, you can also see along the sediment kind of little pellets that are fecal matter of other things that are crawling over the sediment. Oh, really nice, beautiful, beautiful, beautiful. You can see a brittle star moving along the bottom. Okay, beautiful image of that sieve plate right at the top. I'll tell you, this looks This is, and you can see that fringe all along, that kind of like that corona, the fringe all along the top are silica spicules. Some of the sponges, these skeletal elements. Beautiful. Yeah. Very nice. Right. And it looks to me that what those really long, what I thought were kind of filaments that were sticking out at the bottom, might actually be really long spicules as well. So this is a beautiful sponge. Yeah, and down. Let's just take a look at that. Yeah. Oh, wow. How cool. Yeah, really long silica spicules. And you can see a little shrimp that's swimming around, probably picking off some of the organic matter that's settled on the spicules. And those kind of white masses that are inside the sponge are actually the pair of shrimp that are in there. So very cool. Really, really nice.
Ready?
Yes, ready.
This,
It'll be.
This is brittle.
It
And
it won't be
floats
real
a lot.
brittle, but you're good at this. You can just get it right at the.
Yeah. Yeah, it's
Yeah,
a
you-
floaty. Let's hope we can get it.
Okay, we're going to go in and collect this now. Where do we want this?
You want the whole thing? Or just pieces? The whole thing or just
Yeah,
pieces?
the whole thing. Of course, the whole thing. I'm going to say that I don't think it's real brittle, but you might prove me wrong. Wow. All right, where are we going to put that? Do you think the whole thing would fit in the quiver or not?
Or no?
Okay. Well,
You have to put it in either
probably.
Firebox 1 is going to be good. But
Yeah, probably.
it has something in one already. I don't know if it can
Yeah, you know what,
close
that's okay.
with it.
We could do.
I could try for
Try.
these quads.
What are you going to try?
For these, if those front
Yeah.
ones, the
Yeah, one of those two. You think it'll fit one of those two?
you could try.
You can try? Okay.
We've got at least 3 shrimp in there.
Jicky. That one. All right. Okay. We have an amazing ROV team, really, really skilled. This is not easy to do. It's not easy to do. And it's, you know, a lot of times these sponges, they're so fragile, they'll get, they'll get damaged during collection, broken. And our team has a really soft touch with that manipulator and doing an excellent job, so thank you. Okay. Keep on moving. Yep, keep on moving towards waypoint 2. Yeah.
Try to continue with that move.
You were able to get some good photos? Yes. Great, thanks. Okay, we're continuing on. We have several students who are participating in this expedition. They've been really great. They've been logging these dives, making really interesting observations in our database. We have a couple students who are our loggers on this portion of the dive. They're making sure that we capture all the information about what we're seeing, and not only what we're collecting, but also what we're seeing. So every time we see something interesting along the bottom, we make something different, an animal, some unusual activity, they make a note in our database. I think we've collected one of these sea stars before. Just keep on going. So a lot of times the sponge that was, we're just going to keep on going. This is a stalk of one of the Hyalonema sponges, another glass sponge that we've collected a few samples of. This one has, in addition to these little anemone-like organisms called zoanthids, it also has stalked barnacles living on it. Here we see another sea star. We saw a halosaurus fish over on the side. Every now and then we're seeing sargassum, so brown alga on the bottom. I think earlier in the dive you saw some images, well maybe not. We saw some images of the surface of the surface of the ocean here where we are off Trinidad, and large mats of sargassum. And for those of you who are living in the Caribbean, you're on the coast, you're quite familiar with that. And a lot of it just will sediment down to the bottom. This is another halasaurid fish. I am not a fish person, so I'm learning this from the experts who are. So there's a lot of sediment in the water column. Some of the things, the other activities that are going on during this expedition are collections of from the midwater, zooplankton, and a little bit higher up phytoplankton, and looking at kind of the connectivity between the midwater and ocean bottom. So, oh, there's a sea cucumber, holothurian. That's one of the animals that benefits from all the stuff, the organic matter that's sedimenting down onto the bottom. Sea cucumbers basically eating mud and taking from it the nutrients that are found in the bacteria and other microorganisms that are living in the sediment, and the organic material that's settling down to the bottom. Again, we're seeing some mounds here, sort of, I'm not sure what exactly it is that's living in underneath those mounds, probably worms. Seeing some more urchins. Not sure what that is. Let's get a closer look at what's in a couple of things in front of us. What is that? Anemone. Oh yeah, keep going. Okay, we can keep going. Anemone. So that seaweed that's settled down to the bottom eventually breaks down, contributes to the nutrients that are in this sediment that things like the sea cucumbers are munching on. Another sea cucumber. It's another stalk of a dead hexactinellid sponge, and it looks like it's got some barnacles living on it. Might have an anemone on it as well. Looks like a little shrimp. So this expedition is aboard the research vessel Falkor 2, which is owned and operated by the Schmidt Ocean Institute. And I really appreciate the opportunity to be able to explore these unknown areas of the world's ocean. So we've been out to sea for several months. We just started our expedition. We left port a couple days ago. We just started our dives today, and we'll be diving until the end of July. So pretty excited to be on this mission. Throughout the expedition, we'll be looking at different habitats. So we decided to explore this area today. And some other areas we'll be looking at kind of shallower areas along, it's called the mesophotic, like reef areas, so shallower, where we'll start seeing some of the animals that you might be more familiar with from if you're scuba divers. this area along the bottom here in this soft sediment is equally important. Oh, there's a comb jelly, a ctenophore. Pretty shrimp. It's important to document the biodiversity of this area and every other area that we're going to be exploring during this mission. We're documenting it using video, using photos, collecting samples that'll be in the museum that can be studied and identified. We're collecting DNA, including environmental DNA. I mentioned earlier, this is another one of the hyalomema glass sponges. You just keep going. Mentioned earlier on that we were also cryopreserving some of the samples that we're collecting. So we're creating what's called a cryobank. We're kind of think of it as a frozen zoo of the animals. We're taking very small pieces of their tissues and we're putting them in little jars, little vials, and we're filling them with an antifreeze, if you will, although they do freeze. It's called a cryoprotectant. So it gently chills, we can gently chill the tissues down to as low as we want to get. This special cryoprotectant that we're using was developed by our colleagues at a company called Cryocrate, and it allows us to cryopreserve or chill these small pieces of tissue without having to use any toxic chemicals and without having to put them in a -80 freezer, although this ship is so amazing. It actually does have a -80 freezer, but most ships don't, and so the ability to be able to put these tissues in a regular freezer like you have at your house is a game changer for us. So we're looking now, that's looks like a sea cucumber. I think we've collected one before. So some of my colleagues are working on sea cucumbers from Iceland and Norway. And some sea cucumbers produce chemicals that can be developed into drugs to treat diseases like cancer, for example. And what they're working on is trying to develop a sustainable way to use these chemicals. And so what we're doing is developing ways to culture the cells from sea cucumbers, from corals, from sponges, from sea squirts or tunicates. And that way, if we can grow the cells in culture and be able to get them to produce those compounds, then they don't need to be harvested. So none of the work we're doing on this particular expedition is related to biodiscovery, anything like that, but I just wanted to point that out. It's an interesting fun fact. Well, it's a fun fact for me. I have another glass sponge or Hyalonema stalk with zoanthids all along, so there's little colonial anemones along the stalk, and a really pretty solitary anemone right attached to the top of this glass sponge stalk. I wish I could tell you what's making all of those mounds, but I honestly don't know. Seeing quite a few of the urchins that are have those really, like those sacks filled with a fairly toxic compound. And if you've ever gotten stabbed by one of the spines, it's really painful. Speaking from experience. Every now and then you see these long trails along the sediment, tracks of some animal crawling along. It's another hexactinellid sponge, Hyalonema. So we've seen quite a few of these. So we're exploring off the coast of Trinidad and Tobago. So it's there's a large area around Trinidad and Tobago that has not been explored. So that's the main objective of this mission, is to really explore these unexplored waters. Less than 0.001% of Trinidad and Tobago's deep waters have been explored. And at the even greater depths that we'll be getting to later on in this mission, they haven't been explored at all. We discover new species all the time, even in areas where we've explored really often. And so you can just imagine in these areas here that haven't been explored at all, we're pretty excited about the potential to discover some new species. It's been estimated that more than 70% of the deep water species here in Trinidad and Tobago remain to be discovered. So that's pretty exciting. And so we're trying to get some representative samples of each of the new things that we see, and they'll be here and deposited in the museum here at the University of the West Indies and available for scientists to come and study. It's another one of these glass sponge stalks that has barnacles, stalked barnacles growing on it. So as you can imagine, here on this sediment, lots of sediment, not many areas that are hard enough to settle on and attach to. So anything that presents any kind of a hard substrate is settled pretty quickly. So that's why you see a lot of things that are growing on these glass sponge stalks. That was a ctenophore comb jelly that just floated by the camera. So as I mentioned, that the hosting organization is Schmidt Ocean Institute. We're on the research vessel Falkor 2. Participating institutions, there are many. The lead institution is a nonprofit NGO called Species, and it's located in Trinidad and Tobago, and it undertakes scientific research, education, and advocacy. But other institutions are University of the West Indies, St. Augustine, Institute of Marine Affairs. My colleague, Dr. Megan Conkling, is going to be coming on next. She's from Harbor Branch Oceanographic Institute at Florida Atlantic University in the United States, the University of Calgary in Canada, Dalhousie University in Canada, and the National Museum of Natural Sciences in Spain. There are a number of other supporting institutions: Ocean Discovery League in the United States, Pure Ocean in France, the Explorers Club in the United States, the Pew Charitable Trust in the U.S., and the Anthony and Tobago Awards for Caribbean Excellence, and our colleagues at CryoCrate who are providing all of our cryoprotectant to us so that we can create that frozen zoo. Currently at 974 meters. I think we started out the dive at, I think, roughly around 1,040, 1,050 meters.
Yeah.
So we've been coming up gradually. It's a very shallow slope. Temperatures 5.39, 5.4 degrees Celsius. Salinity is a little under 35. Under 35 parts per thousand. Sea cucumber.
And just a heads up for our folks watching at home, we are going to be resetting the stream here in the next 5 minutes or so. So if you don't automatically get redirected over to part 2, just keep an eye peeled for that, and we'll see you there. See the shadows on the bottom of the cone jellies. A whole little community on this one. So this is a live sponge, the hyalonema. It's got the zoanthids, and it's got an anemone. It looked like it had a crab on there.
are these nasty, poisonous sea urchins. Coming up on a white sea star. So I'm going to be handing off narration of the dive now to my colleague, Dr. Megan Conkling, and she'll be guiding you through the dive for the next hour.